From the Library of Congress in Washington, D.C.
Good morning and welcome to our conference on stress. The title of my lecture is Physiologic and Molecular Characterization of the Stress System in Health and Disease. I will try to be as simple as possible without being simplistic. And I will start with a saying by Epictetus, who was a Stoic philosopher. So what he had said is, be equanimous and remember not to believe easily. So I hope that everything we say here is evidence-based and solid. My lecture is divided into 4 parts, and I will go over the 3 parts only today. Stress concepts, stress mechanisms, effects of stress on the organism, and finally, this is a part of a lecture that's going to really be used tomorrow on coping with stress. Stress concepts. Let's begin with a saying by Antisthenes. That's another Stoic philosopher. He said that the beginning of science is the visit of names. So let's visit the name stress, the etymology. It has an Indo-European root, which is str. It exists in ancient and modern Greek as strangalion, which means to strangle. Strangle also, the English is coming from the same root. And in Latin is stringere, which means to press tight, and it's used in modern Italian as well. Now, one of the Western philosophers that started thinking of complex systems, and now we are in the era of systems biology or systems medicine, was Pythagoras, who lived in the 6th century BCE. And Pythagoras suggested that the universe is in a certain harmonious balance, and he called it harmony, which balance is frequently disturbed by disturbing forces and reestablished by adaptive forces within the system. His student, Alcmaeon, called this phenomenon isonomia, which means equal division, equal parts. But then, in the end of the 19th century, Walter Cannon, a famous American physiologist, extremely intuitive, called this homeostasis, or steady state, also of Greek roots. So now we have a certain balance in us, human beings, human societies, called homeostasis, steady state, which is threatened by stressors, which can be physical or emotional, and is being returned to the usual balance by the adaptive response. So when we say stress, at least in medicine, and I hope everywhere, is the state of threatened, and in humans, even perceived as threatened homeostasis. Okay, so somebody feels that his homeostasis or her homeostasis is threatened, by definition, he or she is stressed. Let me just very quickly go over the development of the concepts of stress. As I said, we started with Pythagoras, the harmony of the cosmos. We continue with Alcmaeon, which says that health is the equipoise of opposing forces, or isonomia. Empedocles, who talked about the elements and qualities of life, of the universe, which are harmonious. And then Hippocrates himself, who said that this harmony, this homeostasis, is health, and disease is disturbance of this homeostasis. And he was absolutely right. And he also suggested that nature heals the disease, so there is an adaptive response that will bring the balance back. The Stoics talk very extensively about ataraxia. And ataraxia is the state of imperturbability of mind, or equanimity. So the ideal for a Stoic was to reach a state where nothing would really bother them as much as possible. Epicurus, who was one of the best philosophers of the ancient world, also used ataraxia as the imperturbability of mind, but he also talked about aiponia, which means absence of pain, and hedony. And in hedony, he didn't mean, eating or having sex or anything else. He meant this state of pleasure, of bliss that one has when one is equanimous. He also talked about eustathia, which means eustasis, good state, and the Greek name Eustathios, very common, which means somebody who is in good balance. Thomas Sydenham, the English Hippocrates, in the 17th century, suggested that symptoms and signs of a disease arise also from the reaction of the patient's system. And indeed, as you will see, this is what happens when somebody is stressed. The reaction, which is the hormonal and other mediators of the stress response, are causing the damage. Claude Bernard, of course, talked about the milieu intérieur, so the homeostasis of our internal environment. Walter Cannon talked about homeostasis and stress. He also talked about bodily responses to emotions and coined the term fight or flight reaction. Now we know there's a third reaction, which is a freeze reaction during stress. Either way, you either have to fight or flight or just pretend you're dead. Hans Selye, the last of the great figures in this area, described the general adaptation syndrome or stress syndrome. He said that it doesn't matter what kind of stress you're in. If it's chronic enough, it produces the same manifestations, regardless of the original stressor. And he also talked about disease of adaptation, and Phil Gold and I believe that a lot of human pathology are diseases of adaptation, stress disorders. And he also talked about bad stress, distress, and good stress, eustress. And that's also correct. There is bad stress and there is good stress. Now, the fact that there is a balance in our body was mentioned initially by Epicurus. I just did not go over that because I already mentioned it. But there was another physician, Asclepiades of Bithynia, who suggested, and I think I find this amazing, the human body consists of molecules that are made of atoms and void spaces. That's exactly what we are. And diseases are caused by alterations of form, position, or free flow of a patient's molecules. Okay, so he predicted molecular disease, genetic disease, and so forth. Now, since we're talking about complex system, and stress is a concept that's tightly linked to complex systems, let me mention that humans are the most complex system we know of. And the reason we're so complex is because we have not only 23,000 genes and 3 billion bases in our DNA, but also we have 100 billion neurons in our cortex and 10 to the 18th power synapses in our brain. So if you calculate the complexity, it's something amazing, more than the galaxy. Now, we consist of units, which can be molecules, genes, cells, etc. Networks, because these units can form networks, and the stress system is such a network, basically. We have programs, and we have in our body hardwired several programs, and the stress program is one of them. And finally, we have systems within systems within systems, and it goes like this. So what are complex systems? They're self-organizing, they have multiple interactants, they adapt through feedback loops, they are resilient to perturbations up to a certain point, and they have emergent properties. So as to say, for example, from the planetary system emerged the biosphere on Earth, and from the biosphere emerged life, and from life, man, and from man, intellect or civilization. So there's a series of emergent properties that basically talk about complexity. And here is such a complex system, and what humans are systems within this complex system. So we are small complex systems within a larger complex system, which is the biosphere. And our own complex systems interact with each other, and that's what's called empathy. Interestingly, and that's mostly for physicists, we're open thermodynamically as a system. We have, however, organizational closure. Oops. Excuse me. We generate and sustain a relation, an identity under precarious conditions, and bring forth a meaningful environment. And some basic stuff in us is the will for survival of both the individual and the species, and that's an end on its own. Now, we also form a very complex system as human societies. And here I mention the mirror neurons and the spindle or phonoeconomon neurons that are responsible for the feeling of empathy. In other words, understand how somebody else feels emotionally. And Charles Darwin was the first to talk about the beginnings of empathy, which he considered the love of a mother to a child, and vice versa. That's the beginning of empathy. You start caring about an individual, and that is extended to the partners, to the siblings, cousins, society overall. So what he had said is that any animal, whatever, endowed with well-marked social instincts, the parental and filial affections here included, would inevitably acquire a moral sense of conscience as soon as its intellectual powers had become as well or nearly as well developed as in man. So Franz de Waal, in a beautiful article in the Annual Review of Psychology in 2008, suggested that moral instincts had their beginnings in circuits of emotional contagion or empathy. And indeed, that's the way it seems has worked. Now, another scientist from Harvard, Chrousos, has an evolutionary idea about the cosmos and how we move into ever-increasing power densities. And just remember the concept that as we develop, oops, I'm doing something wrong. No, unfortunately, it doesn't work. But it's okay, I can explain it. The power density is on the left, and time in years to several powers is the abscissa. And what you see is that as the world becomes more and more complex, and that's the power density increases, things are happening much, much faster in lesser time. And right now, with societies, we're at the highest possible level of power density. Now, both Walter Cannon and Hans Selye used the Hooke's law of elasticity to make people understand what is meant by stress. And what is that? If you take a metal rod and press it in the middle, the rod will bend, and the more power you put, the more it bends. So if you express that as an equation here, you'll find that up to a certain point, it's a straight line. So you take the weight again, or the power, and it goes back to normal. So that's homeostasis. Nothing bad is happening to you. But beyond a certain point, which is called the yield point or tipping point, the system starts breaking down. And it never goes back to where it was before. And that is a state which corresponds to human chronic disease and has been called allostasis, or more correctly should be called kakostasis. It's a bad state, dynamic state, that results in earlier death for the individual. And of course, there is a fracture point at the top where it's, the organism is broken down completely, death. Now, let's say somebody's in a normal basal homeostasis and something happened to that somebody. So it's a straight line, we're in our homeostasis, and then a stressor occurs, which can be distress or eustress. And then the adaptive response kicks in, and the individual goes back into its normal homeostasis in the middle. So that's eustasis. Now, some individuals will never go back to their normal state. Example, PTSD, post-traumatic stress disorder. They're not the same people afterwards. So this is deteriorated homeostasis or kakostasis. And of course, there are people who benefit from stress, especially eustress, such as exercise. And their curve goes even higher. And we've called this hyperstasis, a state that's well above, somebody's homeostasis. And then, as we really don't realize all the stressors that are around us, such as noise, daily hassles, and so forth, we don't realize that we're stressed. But some people are negatively affected for a long time, so the line goes down, and some people are affected positively for a long time, and the line goes up. Recently, I saw a beautiful study comparing New Yorkers to people in a smaller town. And basically, New Yorkers had higher cortisol levels. And I can understand that. Their line was above the usual homeostatic line. Then another important concept when dealing with all of these complex systems, especially the homeostatic mechanism of a complex system, is everything in moderation. And that's how these systems work. They work best somewhere in the middle. You see an inverse U-shaped curve. So when we're in the middle, we operate the best. When it's too low, it's in kakostasis, at deficiency. When it's too much, it's in excess, also kakostasis. So we have to be somewhere in the middle. And I'll give you the example of arousal. If you're under-aroused, you don't feel well, and you don't function well. If you're properly aroused, you do your best. And if you're extremely aroused, you have anxiety, and you don't operate well. And that's true for all the homeostatic systems in our body. Now, human stressors can be anything that will disturb our homeostasis. And I start from daily hassles, life transitions, the life of a woman, for example, with its various transitions, natural and unnatural catastrophes, starvation, excessive nutrition, deficient exercise, excessive exercise, obesity, job loss, downsizing, loss of control, bereavement if you lose somebody you love, taking care of sick patients. And here we have we can have pathologic empathy, which is quite stressful for the individual, or unprincipled compassion. We'll say a few words about that. Then addiction, use of toxic substances, all kinds of inflammations, traumatic, infectious, autoimmune, allergic, anxiety, depression, and personality disorder from the psychiatric point of view, and even sleep deficiency nowadays, as our society sleeps 2 hours less a day than these people in the 70s. What is empathy, that I mentioned already several times? Empathy means feeling inside somebody else. N means in pathy. And there is also sympathy, which we use when we feel bad about somebody and we sympathize. And apathy, where we don't feel anything. Compassion is sympathy plus action. And principal compassion is beneficial or at least harmless for both the caring and the cared. Okay? So unprincipled compassion is very stressful. And for physicians, nurses, and other people who take care of people, that's very important. So you have to be compassionate, but you shouldn't damage yourself or the patient in this context. And the other thing that's important is that all of these stressors, when they occur together, they're synergistic. They produce a lot of disturbance if you add them up. And let me just say that there are critical periods of life, such as the prenatal life, childhood, puberty. And remember that the human brain ontogeny is complete at 25 to 27 years. So the final connections of the prefrontal and frontal lobe to the rest of the brain are complete at 26, 27 years. So during these critical periods, hormones can have organizational effects. That means effects that are there forever. And such hormones are the stress hormones, such as CRH, which comes from corticotropin-releasing hormone, glucocorticoids, such as cortisol, as well as sex steroids and cytokines of the immune system. These hormones can have permanent organizational effects in the body. And it's interesting that Leo Tolstoy in the 19th century wrote, and he's absolutely right, "From the child of 5 to myself is but a step. But from the newborn baby to the child of 5 is an appalling distance." And why is that? Because at 3 years of life, we have the maximum number of synapses in our brain. And after that, they start going down. The circuits that remain present and alive are the ones that are used. The ones that are not used are pruned, taken away. So depending on the environment of a child at this important age, 0 to 5 years of life, will be there for that individual forever. Now, what are the homeostatic systems that we have in our body? Well, we have the amygdala, which controls fear and anger. The mesocorticolimbic system, MCLC, which is the reward and punishment system, feeling well when you do good things, feeling bad when you do bad things. The stress system, where I'm going to concentrate. The metabolic system, the immune, the fatigue and pain system, wakefulness and sleep, very important, and the clock system. And in fact, the stress system influences everything. And at this point, I'd like to mention again the prefrontal and frontal lobe, which serves higher functions for us, such as interpretation of the environment, social cues, problem solving, planning for the future, and proper control of impulses. So this is really what makes us human. And in fact, the prefrontal and frontal lobe, the logos, or the logic, are our best homeostatic system that has allowed us to survive and prevail on this planet. Now, let me go to stress mechanisms. The stress system that I'm talking about is a heuristic concept. In the brain, in the hypothalamus, we have two nuclei that produce CRH and vasopressin. These are the two central stress hormones. Those go to the pituitary, they cause secretion of ACTH, corticotropin, and corticotropin then goes to the adrenals and causes secretion of cortisol. The other part of the stress system is the locus coeruleus, that's in the brainstem, the back of our brain, and the norepinephrine system. And the locus coeruleus and norepinephrine system are responsible for arousal and for sympathetic and parasympathetic activity. So the peripheral component of the LC&E system is the sympathetic and the parasympathetic system, and of course the adrenal medulla that produces epinephrine. Now, are these central systems communicating with each other? And the answer is yes. They innervate each other. So you have fibers, nerve fibers from the CRH in the hypothalamus going all the way back to the brainstem to stimulate the brainstem neurons that secrete norepinephrine and vice versa. Norepinephrine stimulates CRH. So we have a positive feedback loop, and in our organism there are many positive feedback loops, but the ones we have are extremely important for survival, and that's one of them. Now, if you don't need to remember this, just remember that this is a complex system. It interacts with other systems in the brain, and it has its own feedback loops. There are negative feedback loops that control it. And one of the most important feedback loops is from the GABA benzodiazepine system. If you give Valium, that system is suppressed. And that's because GABA regulates the system negatively. Also, there is interactions with the cholinergic system, the serotonergic system, with oxytocin, and of course with the mesocortical mesolimbic system and the amygdala and the hippocampus. So the stress system sits in the middle in the body and affects development, behavior, growth, immune system, the clock system, and wakefulness and sleep, very important, the reproductive system, and finally participates in our aging. So it's a very central system which helps us but also can damage us. And if I put it again in a simpler way here, I'll just mention that here on the left is the HPA axis, hypothalamic pituitary adrenal axis, and on the right is the sympathetic system. And you'll see that in the amygdala, the center for fear and anger, the neurotransmitter is CRH, the same one that's in the hypothalamus. And actually, there is a second positive feedback loop there, where the amygdala stimulates the LCNE system and vice versa. Also feedback important, and also something that needs to be controlled. In the periphery, we produce not only cortisol, the F that you see there is cortisol when we were stressed, but we also produce norepinephrine and epinephrine, and also produce from the same peripheral nervous system CRH, the same one that's in the hypothalamus. And in the periphery, CRH is pro-inflammatory, acutely pro-inflammatory, causes inflammation. And then both norepinephrine and epinephrine stimulate during stress a hormone of the immune system which is called interleukin-6, which is a pro-inflammatory hormone. So you realize that when you're stressed, you produce not only hormones that you know that are stress hormones, but also others that you don't suspect. Now, I have this, again, it's for me to just explain to you the general principle. The stress system interacts with these 4 other systems in the brain, and all together they form a circuit. So if you affect one part of the circuit, you affect the others. And here I have the example. The stress system is at the bottom, so it's secretion of cortisol, catecholamines, and IL-6. And then there is a positive feedback loop with the amygdala, stimulating each other. There is positive stimulation by stress of the reward system, the MCLS, reward system. So whenever we're stressed, we stimulate reward, because otherwise we wouldn't do anything. We'll abandon any effort. Okay, so we need to stimulate reward during stress. And then we have the hippocampus, which is important for intermediate memory, which is also the brake of the stress system. So the hippocampus sends this GABAergic inhibitory effect on the stress system. So you can see that if you damage any of these 4 parts of the system, you will damage the others, because they're so highly interlinked with each other. And here, too much to see, but I'll just tell you this. We know exactly what stress is doing to our reproduction. It causes hypogonadism. In other words, it decreases our reproductive capacity from libido all the way to the levels of the hormones. It inhibits growth, so it's stressed child, for as long as it's stressed, stops growing. It affects the thyroid system, which is responsible for the burning of fuel in our body, so it suppresses that. And finally, it has a very complex interaction with the immune system. So when we have an immune response, the stress system is activated, whereas when the stress system is activated, the overall effect is anti-inflammatory. So when we're stressed, our immune system doesn't work very well, because innate immunity and adaptive immunity, part of it, are suppressed. And I'm coming back to inflammation as stress. Inflammation is extremely important, as you'll see soon. We have recognition receptors in our cells, all of our cells, which actually, when they recognize a foreign substance or a substance that is injurious, it activates special proteins inside the nucleus that cause the cell to be inflamed. And acutely, this is an adaptive response. Chronically, it's not adaptive. You have chronic inflammation in a cell, and it's actually carcinogenic. Now we know that chronic inflammation also is important for oncogenesis, cancerogenesis. Now, these are the various inflammatory injurious agents, from microbial products to intracellular molecules to denatured molecules. For example, oxidized molecules produce inflammation in our cells. Adducts, toxins, it doesn't matter. Anything that's injurious will activate this system. And here I have the example of lipopolysaccharide, which is part of the wall of the gram-negative bacterium, which actually, through specific receptors, pattern recognition receptors, activates inflammation in the cell. And once that happens, then you start producing inflammatory cytokines. And the first one that's produced is TNF-alpha, tumor necrosis factor alpha, the second IL-1, IL-6. It's a cascade of events. And the end result is these cytokines circulate in the blood. And what they do is that they activate the entire immune response in the nucleus. So this transcription factor, NF-kappa B, you can't see it very well, doesn't matter, goes in the nucleus when activated and activates all the genes that are responsible for inflammation. And in fact, some of these, the products are the inflammatory cytokines that come back and activate the cell again. So this is a third important positive feedback loop. And there is one more in the reproduction. 4 extremely important positive feedback loops in our body. Now, whenever we are inflamed, there are 4 programs that are activated in our body and brain. The first program is called sickness syndrome or sickness behavior. It's how we feel when we have the flu. The second is the acute phase reaction. You recognize, physicians look at C-reactive protein, that's an acute phase reactant. The pain and fatigue neural afferent program, and finally the stress program. So you get the first 3, and once the cytokines go above a certain level, then you activate the entire stress program, which is responsible for helping inflammation on one side, but also blocking inflammation on the other. Absence of cortisol means increased inflammatory responses. Cortisol has to be just right to prevent inflammation from overshooting. So this is 4 different networks and 4 different programs that are activated. There is overlap among themselves. However, one should see them as highly interlinked. Now, we know that in sickness syndrome, when the program of sickness is activated, in our brain, cytokines are either crossing in the brain or produced locally in the brain, or both. And the end result is to have this sickness behavior. We know exactly what happens to the various neurotransmitter systems and so forth. It's an adaptive response. When you're stressed by inflammation, you have to stay in bed and wait until you get better. You don't want to waste resources anywhere else. And that's exactly this program. So let me now go to effects of stress on the organism. And let me mention that time is extremely important when it occurs. Okay, especially if it occurs during the critical periods of life, prenatal, first 5 years in adolescence. Second, acuity. Acute stress can trigger some pathologic responses, and chronicity. And unfortunately, this didn't come out, but it doesn't matter. During critical periods of life, when you're stressed, let's say in utero or the first years of life, but in entire life, but here is more, early on, you're much more sensitive. You get the so-called epigenetic changes. In other words, you change forever the activity of genes by methylating the regulatory areas or the structural areas and so forth. So somebody who's exposed to stress early in life is bound to have changes that will be there for the rest of their life. That's called epigenetic, and it's the basis of all these changes. So now let's go to acute stress and then chronic stress. Acute stress. An acute stress response can cause asthma, precipitate asthma, eczema in the skin, or urticaria. It can precipitate migraine headaches or tension headaches. It can precipitate gastrointestinal pain, and actually in children and women, this is the most common psychosomatic complaint. That they go to the doctor, they're anxious, and anxiety is somaticized as abdominal pain. It's basically the stress system stimulating the GI system in the abdomen. You can have a hypertensive episode, and if a host is compromised, somebody with atherosclerosis, for example, older age and so forth, one can have a cerebrovascular accident or even die during stress. Also, one can have a panic attack during stress. Also, we can have cardiac ischemia, an NMI, arrhythmia, death again in a compromised host. And finally, in people who have the right genetics, you can precipitate a psychotic episode. Now, this is what stress is doing to inflammation. Nerve cells from our peripheral nervous system produce CRH, which is the dark area. So if you look at an inflammatory site, it's just full of CRH. And what does CRH do there? It degranulates mast cells. These are cells that sit locally in our tissues, and when they're activated by CRH, they secrete cytokines, histamine, etc., and influence local inflammation. So it's a key factor in the way acute stress influences the inflammatory response. And here is how stress influences the GI tract. On one hand, CRH inhibits the vagus nerve, and we have decreased gastric motility and indigestion. Typically, you feel full if you're stressed, you don't digest. And then on the other side, CRH activates through the LCNE neuron, the sacral parasympathetic system, to increase colonic motility, and that's irritable bowel syndrome is within that context. Now, what are the chronic effects of stress system malfunction? Let me go first to behavioral and say that early effects hit right at the logos and at the self-regulatory system. People who are stressed early in life frequently are not able then to regulate their impulses. So they're impulsive, they don't realize the long-term consequences of their acts and do negative things, and that gets them into trouble. Fear and anger is activated, that system, and reward and punishment is activated. Also, the cardiovascular system, the metabolic system, the immune system, pain and fatigue system, and sleep, they're all affected by stress. And let me here mention what are the chronic effects of stress system activation. It causes anxiety, depression, addiction, antisocial behavior, psychosomatic disorders, fatigue, and pain. It causes loss of weight and poor growth, or it causes obesity and a metabolic syndrome, and I'll say a few words about this. It's also associated with smoldering, low-grade systemic inflammation, immune dysfunction, atherosclerosis, cardiovascular disease. Then osteoporosis, chronic stress through cortisol and IL-6 causes osteoporosis in our bones. It also causes premature aging of all vulnerable organs, including the brain, that's neurodegeneration, and the skin. And finally, increases vulnerability to infections and cancer. So if you look at these disorders, you can see that most of them are the so-called chronic non-communicative diseases. These are the diseases that curtail our life expectancy. This is the problem in the world now. It's not the infectious disease, at least in the Western world, it's the chronic non-communicative disorders. And they're all influenced by stress. And again, I'm going back to this circuit that I showed you earlier. And we believe that somebody, when they're stress hyperresponsive, they have a large stress system that's activated robustly. They have a large amygdala, so fear, anger, etc. They have decreased hippocampus, and actually you can see that on MRI. You can measure the size, and it's decreased in stressed people. And a decreased mesocortical and mesolimbic system, a reward system. So this is a perfect combination for somebody who is stressed, anxious, doesn't feel well, cannot control their impulses, and so forth. Now, what happens during chronic stress? During chronic stress, if you measure cortisol and IL-6, etc., you'll find that's elevated. And for cortisol, we missed the boat for many years because we saw that when somebody is stressed, they produce not much more cortisol, a little more than normal, okay, every day though, for years. But they produce it during the evening, okay? So if you look in the top curve, the interrupted line is somebody who is chronically stressed. Cortisol is high in the evening at a time when it should be undetectable in somebody who's chronically stressed. And when that happens, is recently found and published, is when the sensitivity of our tissues to cortisol is elevated in the evening. This was known by physicians who treat with glucocorticoids. But now we know exactly why that happens. The receptor is deacetylated in the evening, and it's much more sensitive. So even small elevations of cortisol over time, in many years, it will cause the effects of hypercortisolism, which is, you may have heard it, is Cushing syndrome. And also, the other thing we need to remember is that during stress, we produce cytokines, such as IL-6. So non-inflammatory stress is associated with hypercytokinemia, high immune function proteins, cytokines, which stimulate inflammation. And then I'm going to say a few words about obesity, especially visceral obesity and aging. If you plot somebody's body mass index, okay, and against levels of interleukin-6 or TNF-alpha, these are two very powerful inflammatory cytokines, you get a positive response, a positive correlation. So the more obese you are, the more cytokines you produce. Therefore, the more systemic low-level inflammation you have. And that goes together with the elevations of IL-6 that occur during chronic stress. So if you're obese and chronically stressed, it's a bad combination. Now, in this slide, I'm just summarizing everything I said. Basically, depending on our genetic variation, our genes, and our developmental history, how much stress we were exposed early in life, and how much stress we are exposed to now, currently, and it doesn't have to be real stress, it can be perceived stress, and on the basis of what we eat, nutrition, and on the basis of our age, we hypersecrete cortisol, norepinephrine, epinephrine, and interleukin-6. We hyposecrete other important hormones for growth, and the end result is to collect visceral fat inside our abdomen. Why is that? Because cortisol stimulates glucose, and glucose stimulates insulin. And insulin together with cortisol are very potent growth factors for the fat in the abdomen. This is the fat that causes the damage, okay, the visceral fat. You may have heard the word also insulin resistance, same thing. And also somebody who's chronically stressed, lose muscle and bone, lean body mass. So the end result is to have insulin resistance, dyslipidemia, dyscoagulation of our blood. For people who are genetically sensitive to polycystic ovary syndrome, women, they can develop PCOS, osteoporosis, of course, sleep apnea. And the key thing is that why we live briefer lives if we're chronically stressed is because our cytokines and other hormonal mediators influence our endothelium, the layer inside our vessels. So if you activate the endothelium, then that attracts white cells there and start formation of plaques and so forth. That means atherosclerosis and cardiovascular disease. And in the United States and the rest of the world now, we know that the curve of body mass index of Americans has moved to the right. So we're now much more obese than we used to be. The red curve is moving to the right over a period of 30 years. And now we know that 1/3 of Americans at the age of 55 have metabolic syndrome, the full metabolic syndrome, dyslipidemia, hypertension, etc., diabetes type 2. And 1/3 have partial metabolic syndrome, and only 1/3 are healthy. And I believe that to a great extent, this is due to chronic stress that hasn't been managed well for a long time. Sleep apnea, that also depends on the amount of visceral fat in the abdomen. There's the formation of plaques through activation of the endothelium. And finally, let me say a few words about stress of our cells, cellular stress. Every cell, when we're stressed, of our body is stressed as well. And there are 3 types of stress that are pertinent to this. One is nutritional stress. If you eat too much or too little, so too much food, too little food. Inflammatory stress, injurious agents, and oxidative stress, production of oxygen radicals in our cells by the mitochondria. So in fact, nutritional nowadays, it's insulin resistance, what I mentioned before. Inflammatory is through specific agents inside the nucleus of the cell, and oxidative through the mitochondria. When our catecholamines are up, epinephrine or epinephrine, they stimulate mitochondrial function, so we produce oxygen radicals. And chronic cortisol elevations cause biogenesis of the mitochondria. So you have more mitochondria in somebody who is stressed and produces more oxygen radicals. Now, how about our telomeres? Are they affected by stress? Telomeres are little pieces of DNA, repetitive, that sit at the edge of our chromosomes and protect our chromosomes. And in somatic cells, there's a limited number of duplications of cells that can occur, up to about 30 duplications. And every time the cell duplicates, it loses some of the telomere length. Now, look at this study. It shows that the more obese you are, the more metabolic problems you have, the smaller your telomeres become. So in fact, now we know, and Dr. Wadwa will say more about this later on, that stress decreases telomere length and therefore directly influences our age, our aging. And I've been impressed at how quickly people who have chronic stress age, such as parents of children with autism, for example. I've seen it in my department where you see them a few years later and they look much older. And that's because day and night they have a chronic stressor to damage their body. So stress, metabolic syndrome, and depression, Dr. Gold is going to talk about that, are tightly linked to each other. They go together very frequently. And inflammation, sleep disturbances, and fatigue is part of all three of these conditions. And we know why, we know the mediators that are affected. And actually, I just mention it as an interesting thing, drugs that improve our life expectancy belong to either anti-stress drugs, such as antidepressants, beta blockers, converting enzyme inhibitors, angiotensin II blockers, anti-inflammatory agents such as omega-3 fatty acids and saturated fatty acids, PPAR gamma agonists, and antioxidation molecules such as phytoestrogens and sulforaphane, and insulin sensitization agents, diet, exercise, use of metformin, etc. And finally, anticoagulation, antiplatelet agents. So we can now prolong the life of an individual by using these drugs, and we know that is happening in the United States and elsewhere. So let me finish here and open the floor for discussion, and we have enough time for that, with something that Aristotle wrote. He wrote that the soul suffers when the body is diseased or traumatized, while the body suffers when the soul is ailing. And indeed, this is true. Thank you very much. Sure. Hereditary sides of stress, yes. I heard it. So you're interested in the hereditary side of stress. The question is, how hereditary is the stress response? Yes. And the answer is, to a great extent, it's hereditary. In other words, there are some people who have the right genome, right combination of genes, that make them more responsive to stress than others. This doesn't mean that it doesn't change. It's reversible. It changes depending on our early life. So you can take, Dr. Jerome Kagan had done these studies. You can take a child who is very stressed, a so-called inhibited child, put them in the right environment, and they become outgoing and productive and so forth. So now we tend to think that heredity is important, but environment appears to be more important than heredity. Dr. Rosenthal.
Yes, firstly, thank you for that very, very nice presentation, very enjoyable. Question, epigenetic effects of stress. Are they hereditary themselves? Is there any evidence that the epigenetic changes can be transmitted?
Let me tell you that both genetics and epigenetics are influenced in a major fashion by stress. So our species went through stressors, and we are who we are genetically. We have the tendency to get obese, to get anxious and so forth. This is because we went through those bottlenecks of genetics. But then epigenetics, the majority of epigenetic effects are not transmissible. They're for the ontogenies, for the lifetime of an individual. However, some of them do get transmitted. For example, you may have heard the famine of Holland, where now for 3 generations that they found that children are more prone to metabolic syndrome. 3 generations, which means they obtained this epigenetic vulnerability that lasted long. So the answer is that some of the epigenetic effects do get transmitted through generations, but very few.
Second question was that I saw the correlations between stress and telomere
the telomere effects, I think it was a body mass index or
Yeah,
some,
body mass index or
some
adiponectin.
measurable.
Yeah, yeah.
The graphs you had up there
Right.
seem to show about a 0.3 correlation, which would suggest like a 10% variance
Right.
accounted for. Now that's not trivial, but it's not huge. So
Thank God, yeah.
Thanks
It's
very
not huge,
much.
but it's there. And of course, it depends on the amount of stress that somebody is exposed. This is a general population that we saw.
Particularly interested in the implications of what you said for circadian rhythm dysregulation. It was implicit, but I wonder if you could say a little more. We're used to thinking of stress equaling cortisol, and what you point out is cord elevations may not be all that high, but when they occur is very important. So the deacetylation of receptors, the increase in the evening, you mentioned sleep apnea, but I wonder if you could say a little more about the effects of stress on dysregulation of circadian systems.
Stress doesn't really change the phase of the circadian rhythm, but leads to production of hormones such as cortisol that are slightly elevated in the evening hours. Now, the glucocorticoid receptor is highly acetylated by clock. This is the transition factor that regulates the circadian rhythm, which we have in all of our cells, not just in the brain, everywhere. So in the morning it's acetylated and it doesn't sense cortisol very well. In the evening it's deacetylated, most likely by an enzyme called sirtuin. I don't know if you've heard of it. That's where resveratrol presumably acts from red wine. So sirtuin deacetylates the receptor, and in the evening it's very sensitive. So if you have chronic stress, or if you travel across time zones frequently, or if you work shifts alternating between 3 or 4 weeks and so forth, the end result is that your tissues are exposed to too much cortisol. And you develop everything I said from that, including visceral fat accumulation with all that it entails.
My field, but so this may be a naive question, but I'm getting the idea that a lot of stress is not very good. Is there the corollary where too little stress is also not good, and what does that imply for the middle, the mean?
I'm glad you're mentioning it. Now, and it's very recent, we find that not having stress at all is not good. Yeah. So eustress that Hans Selye talked about is what we need. So some stress, not excessive to go beyond our capacity to deal with it, but not missing on the other side. So again, we're back in everything in moderation. We need some stress to survive. Too much is bad, too little is bad.
Do we know enough that we can educate at those levels?
Unfortunately, everyone is different. Each one of us is different, so we have to define ourselves. Generally, if you're excessively stressed, you know it. You become dysphoric. If you say, I feel stressed, you are stressed by definition. So your own reward system will tell you that you're doing something bad, that you need to change.
It seems like every month we're having new findings on the impact of the microbiota, everything colonizing everything from the gut to the skin. What do we know about the impact of stress upon these systems and sort of the feedback loops there?
I don't know that we know anything about it. And as you probably mentioned, the microbiome is extremely important for us. It's a symbiotic and helps us more than it damages us. And I'm sure in chronic stress, there will be changes because the gut function is affected, glucose function is affected. There are several functions that will be in interaction with the microbiome in the gut or the skin. So I believe there's going to be a lot of effects, but nobody, to my knowledge, has studied it yet. Dr. Smith?
Well, yes, well, very nice presentation and very complete. I just want to comment a little bit about the role of diet, because there is no question that the current diet particularly in Western cultures, is extremely high in omega-6 fatty acids that are pro-inflammatory. And there is very good evidence that the omega-6 fatty acids, for example, shorten telomeres. And the telomeres are definitely shorter in patients who have coronary heart disease. But if they have a balanced amount of omega-6 and omega-3 fatty acids, they have less severe disease and the telomeres are not as shortened. Now, since the current diet is very high in omega-6 fatty acids, in essence, it is a pro-inflammatory diet. And they showed very clearly the role of inflammation and the effects on the stress system. So that when the studies are being done, it seems to me it will make a difference, the response of the individuals or populations on their background diet. Because there is very good evidence in humans that as you lower the omega-6 and raise the omega-3 and balance the ratio, that you have lower interleukin, lower TNF, you suppress nuclear factor kappa B, so that this whole concept of studying stress, I think it's very important to consider the population, the diet of the population, and maybe somehow you need to find a way to evaluate the state of inflammation of the individuals or populations. Because the results could be very different.
Yes. As you probably saw, I had omega-3 in the list there.
Yes,
and diet.
I did notice that. But and exercise, of course,
so
is very
exercise
important.
and proper diet are key.
I just want to make a comment, the microbiome, because this is a whole new field of metagenomics, and there is very good evidence that the population of bacteria varies with the type of diet that we have, and then they go through the mucosa and they influence the inflammatory response in individuals. No question about that.
Dr. Rosenthal.
I just want to mention that Dr. Robert Schneider over here and I are going to be talking tomorrow about the effects of transcendental meditation, and not to sort of, give too much of the game away. I think it's relevant to this particular point over here, because what you're saying is that stress is triggering a tremendous amount of physical and mental trouble, and that we want to try to modulate that stress in every way possible. And what we'll be showing, Dr. Schneider on the physical level and me on the psychological level, is that this form of meditation is well documented to be a modulator of that stress, and the actual end results, the toxic end results, have actually been documented to be decreased. Thanks.
I've seen those studies and I'm convinced, basically, that it works.
Yeah, I was just going to mention, I'm actually going to present some brain imaging data on mindfulness meditation in a moment. But Alyssa Eppel in UC San Francisco, following up on telomeres and aging and stress, has actually demonstrated that even as little as 2 months of daily meditation, which is actually very little, reduces the aging effect, probably mediated by reducing stress, as indicated by telomeres shrinking more slowly. So there's clearly actually empirical evidence that what we choose to do, i.e. mental training, influences our level of anxiety and reduces the speed of aging, even in telomeres, through telomerase, the enzyme. Yeah.
Good.
So there's a rich story.
Well, I think it will be very interesting to see what Fabrizio Benedetti has to say later on, because it seems that placebo and nocebo work partly through the stress system. So something you expect to be good is associated with betterment of the HPA axis of the cortisol production, And when it's something bad, like nocebo, it's the opposite. So, and of course, there are other things like the reward system is affected, etc. However, I'm looking forward to listen to that talk because we're going to get a lot of good explanations about what's happening just on the basis of what we expect that happens.
Actually, I was very impressed a number of years ago that someone did a study with on cortisol, but real-time measurement of cortisol, University of Michigan, I'm forgetting his name, but where they had continuous sampling for 3 hours, and they simply asked the person to modulate their moment-to-moment cortisol response over 3 hours by shifting how they were thinking and interpreting. And a very clear demonstration that how we think and how we modify what we're thinking about literally modulates the increase and decrease of cortisol over 3 hours. And that was a very clear demonstration of thought to adrenal gland.
Dr. Rosenthal.
Just to respond to that, there's one review article on the effects of stress on the
Vascular system. And there's one very interesting graph there. They gave the subjects isoproterenol, which, as many people here will know, raises blood pressure, and everybody's blood pressure went up, and one woman's blood pressure went down. And they asked, while she was getting the isoproterenol, and they asked her what she was doing afterwards. She said, "Oh, I did my usual meditation." So it actually counteracted the effect of a potent blood pressure increaser.
Yeah.
Any other comments or questions?
Is there any correlation on who has too much or too little according to culture? I mean, in other words, can you make any generalizations? It's a really amateur question, but about somebody who's not very well informed about the technical medical side, but it raises the question, is there, quite apart from techniques, individual techniques or individual medical treatments, are there any broad correlations in terms of the world's cultures or the principal national or linguistic or however you want to define it? Is there any at all correlation between who has too much and too little stress?
The answer is we expect that there is a correlation, and I can give you, since we have time, the data from a long-term study that was done in Italy by Professor Timio. He followed women from a certain town in Italy for 30 years, and at the same time, he followed women from the same community that went into a monastery, convent, and became nuns. And it's fascinating to see how quickly women in town developed not only obesity, but also cardiovascular disease, hypertension, and many of them by the end of 30 years had died, whereas the nuns has just gotten some obesity, a little bit of metabolic syndrome, no hypertension, and almost all of them survived for a very long time. So just the atmosphere, the environment of the monastery was enough to change the life expectancy of these people and how they manifested. So I believe countries now like Greece, where there is a lot of stress, we start seeing many problems related to the stress response, from a doubling of suicides to a doubling of admissions to the psychiatric hospitals. And I'm sure within the next 2 or 3 years we'll be seeing more as far as metabolic problems, etc. So the answer is the environment is key. And to deal with stress, you either change the environment or you change yourself, if you can change the environment, so that you can cope with whatever there is. And that's the secret, I think. And we'll discuss tomorrow.
I'm interested if there have been any studies on crying.
Not that I know of, but there may be.
Crying as, I mean, there are studies that use crying as a dependent variable. So you show like sad film clips and you show it to people who are have never been depressed, people who have recovered from major depression, and you look at the rates of crying. But I think, what are you actually asking? Crying?
Especially in our society, it seems that women are more allowed to cry than men. And I just think it's interesting to think about how that's handled and what the effects of that are.
Well, it's an expression of sadness, I guess. Yeah.
Yeah, I think that this is, to me, one of the most fascinating, unstudied questions, because as a clinician, we know there's the phenomenon of having a good cry. Why don't you have a good cry and get it out of your system? It's a very, very well known, I mean, most of us have had the experience of crying and then feeling better. And I think it would be fairly obvious that there must be some neuroendocrine function going on when we cry that relieves stress and tension that has, to my knowledge, never been studied. And I know oftentimes when people are depressed, one of their the biggest complaints is that they cannot cry. And I remember having one of my patients who was depressed on an antidepressant, and she went to her father's funeral, and her biggest complaint was that she had been unable to cry. So I think that this is a sort of unstudied phenomenon whereby obviously the sort of exocrine function of crying is probably correlated with some endocrine or neurological functions that have homeostatic effects that, to my knowledge, have never been studied. Thanks.
Let me also mention that it's an opportunity to discuss the fact that men and women have different responses to stress. Generally, women are more sensitive. Estrogen stimulates both the CRH gene and stimulates anabolism of catecholamines. So women have a better stress system, which apparently is very helpful. But also, women develop diseases of the stress system more commonly, more often, such as depression, eating disorders. And the same is true for the immune system. Women have a better immune system, which is less affected by glucocorticoids than the system of men. But again, this is so good to protect them from infections, and etc. At the same time, causes the so-called autoimmune disorders, which is, again, higher as percentage in women than in men.
Hi.
Yes?
Tears are full of neurohormones, said Dr. Gold, and opiates, for example.
Is it more than, let's say, other fluids, such as sweat or saliva? Because you'll find those hormones in all the circulating fluids.
I taught stress management at American University for a number of years, and I had my students bring in studies as part of the course of the effects of stress on each of the 6 areas of health that we study globally. And I'm positive that one of the studies that the students brought in studied tears and the chemical makeup of tears that were released during stressful environments, as well as happy tears, sad tears, and there was a chemical difference. So I wish I could tell you exactly this study, but I mean, we're talking thousands that I've reviewed over the years. So, but I do remember that very specifically because we were looking at the emotional versus the physical versus environmental and community.
So I'm wondering, tears of joy versus tears of sadness.
Yes,
different
chemical
profile.
Yes,
yeah.
It was. Yes, it was a fascinating study. I'm sorry that I can't recall exactly which one it was.
Wow.
I'll be mentioning tomorrow in our study of support groups for breast cancer patients that one of the things many cancer patients try to suppress their fear, anger, and sadness about the disease as though somehow feeling sad about it will make it happen more. Our groups do the opposite, and we're actually able to show that we could reduce their tendency to suppress their anger and sadness and fear, and that was related to long-term less depression and anxiety, and the intervention seems to also have an effect on survival. I'll be talking about that. But clearly, it's very consistent with what you've heard, that helping people directly express these emotions is helpful. It's a good stress management tool. It's not a bad thing.
To know, I'm curious to know how the knowledge of stress and its effect on the disease being actually practiced in clinical medicine. For example, do you see in the near future that maybe there will be bioindicators that can be used to actually diagnose somebody that if you have hypothyroidism, it is due to stress, and so that the management can be accordingly done?
Oh, I'm sure that we will be able to have sensitive measures of the stress response in the next few years. Right now, what we measure is rather crude, and it's not well individualized. It's good to study populations, but not an individual subject. However, knowing the level of stress of somebody would be extremely helpful on how to deal.
Yeah, I would suggest both the biological, but also the psychological, because in many cases, as you already mentioned in your talk, the same exact external stressor for one person can cause incredible reactivity, and for another person who has a lot of equanimity or flexibility, it's benign. So there's a lot that has to do with our psychological filters through which we actually label something as fear-inducing or not. Yeah.
Schneider.
In terms of clinical applications, and you talked about children, there are many school systems now in the US and South America and around the world that are now using meditation programs as part of their education, transcendental meditation and other programs, and this really affects the children's development, as you theorized, on the stress side, but on the positive stress management side, there's improved academic performance and behavior and even health parameters. So this stress knowledge can be used institutionally and for health policy development.
Totally agree.
It's 10:30, so we have a break for 15 minutes for a coffee, and we'll continue.
Thank you. First of all, just thank you very much for inviting me, and I hope that this is really useful, interesting, and provocative to make us all think and reduce our stress. Okay, very simply put, and first I wanted to say thank you to George for laying the ground on top of which this talk can sit, and making my job a lot easier. So I thank you very much. So very simply put, and I should, as a disclaimer, I'm trained both as a clinical psychologist and also as a cognitive neuroscientist, and I do both and more, which you'll see towards the end of this talk. Psychological stress, right? So it's this ubiquitous problem that we all experience and have to deal with. We have no choice. And my work really goes across looking at stress, anxiety, different forms of suffering, looking at the brain systems in which psychological abilities like emotion awareness, emotion regulation are instantiated, i.e. cognitive neuroscience. And for today's talk, I study many things, cognitive behavioral therapy, vipassana, compassion, but for today I'm going to talk about mindfulness. And all with the clear intention of reducing suffering, understanding mechanisms, and optimizing performance, interpersonally, intrapersonally. So the motivation is the reality that there's stress, but more importantly, stress reactivity, and that each of us have our own unique profiles and how we respond to stressors. And the question is, how do we get from stress reactivity that constrains us, makes us less able to function, to the other side of the screen, which is really about resilience, psychological flexibility, biological flexibility. And how do we actually take responsibility in ourselves to train ourselves in skills that make this happen, make us much more optimal human beings. As has already been pointed out, stress, the word stress as opposed to stressors. Hans Selye in 1936, Western scientists, the non-specific response of the body to any demand for change. Special significance that we, our minds, project, and attach to something. And that's very key. And that, of course, it causes anxiety, burden, tension, et cetera, et cetera. But in response, the other side of it is it's really the stressor. Stressors are anything that cause wear and tear, especially in the hippocampus, but in the whole person, on the body's physical and mental resources. As was elegantly pointed out, it disturbs our homeostasis. Hence, I love that you use the word equilibrium. The ability to dynamically know when I am not in a balanced state, and the skillfulness to bring myself back. It can be acute or chronic, external or internal. So what are the top 10 stressors according to large surveys? As you can see, childhood trauma, death of a loved one, divorce, finances, job, health, personal relationships, chronically ill child, pregnancy, danger, and the list goes on and on and on. So this is simply to motivate us to get our act in gear and learn specific skills to be able to deal with these stressors. So as was pointed out, there are incredibly clear gender differences. Shelley Taylor, psychologist at UCLA, and many others have clearly defined that men, in contrast to women, relatively have much more of a fight or flight response. Think of the amygdala. So that's a profile for protecting or defending or saving yourself. In contrast, women, and again, these are generalizations, tend to have a very different response. Tend and befriend. Attend, pay attention, approach, connect. A very different mode than most males. So we males have something to learn from women, given that the contexts have really changed. It's no longer lions, tigers, and bears, but intrapersonal and interpersonal things that causes fear and stress. So my day job is that I work at Stanford and run a lab and do lots of research with incredibly wonderful people. My night job is that I teach and work at Google, which is really refreshing to have feet, one foot in academia, one foot in biotech business. So teach there regularly, and here's some data from 80 Google engineers in Mountain View, just south of Stanford. So I'm going to propose, and again, this is going back to my theme as a psychologist, perceived stress. How we react to things that are stressors are very powerfully influence a whole matrix of other psychological abilities or qualities. So look at this. So these engineers who report high levels of perceived stress report much reduced ability to regulate their emotions, lower mindfulness skills, lower social support, self-esteem, satisfaction with life, emotional intelligence, self-compassion, empathy. Are you getting depressed yet? Or an increased rumination, worry, social anxiety, loneliness. So this is actually, think about it, these are the 20 to 30-year-old, super hyper-intelligent, creative, innovative people who make everything on the internet happen. And they're very stressed in one of the best places on the planet to work. It's a fantastic place to work. People are incredibly internally stressed to be creative. So for this reason, actually, you'll see that we've developed everything I'm about to say has been transformed into a program to help Googlers. And now it's actually going beyond Google. So what's actually happening when we're in the moment? So I'm really into mechanisms. Well, there's something called amygdala hijack. When the amygdala, shown here in red, within milliseconds, perceives something as threatening, real or imagined. Within milliseconds, it kicks in and it becomes activated. Emotion detection, this is important to me. This is personally salient. Get in gear. It immediately pings other areas. The idea of hijack is that the amygdala, which is one of the most interconnected brain regions in the human animal, us, actually, within milliseconds, influences all of the higher order cognitive abilities. The higher functions is the word that George used. So within milliseconds, our ability to focus our attention can be hijacked, disturbed, interrupted, redirected. The ability to think clearly, cognitive perspective taking, the key to empathy, compassion, pro-social behaviors, can be unended or undermined within milliseconds. Decision making, completely influenced by activity in the amygdala within milliseconds. So the amygdala, Wall Street, on the floor, people being driven by their emotions, making decisions to sell or buy, or to take advantage, or to be honest. So the amygdala, within milliseconds, it's influencing every part of our education, every part of our prefrontal cortex, our ability from attention to higher abstract thinking. So as has already been pointed out, especially, and I'm focusing on the amygdala and the hippocampus today, and you'll see why. Within milliseconds, the amygdala is important for increasing or decreasing arousal, for activation of sympathetic nervous system, gearing up and getting ready, behavioral freezing, which is one way to deal with a very acute stressful situation, stress hormones, as was elegantly explained already by George, through the HPA axis, and inactivation of the parasympathetic, which actually helps us put on the brakes. So you lose the ability to put on the brakes. So this is obviously very complex. And as has already been pointed out, one pathway is the HPA, through which our thinking, feeling, decision-making influences our release of glucocorticoids, and specifically in humans, cortisol. And we know that cortisol feeds back right into the hippocampus and other parts of the brain, but especially the hippocampus. And I'm mentioning that because, as shown here in the beautiful little orange or red, is that elegant research has shown that chronic stress leads to chronically elevated hypercortisol and damages specifically the CA3 field within humans. And this is important because it's been related to impaired memory encoding. Some people in acute stress cannot encode and thus cannot retrieve memories, right? So you think of trauma, be it here or in East Palo Alto or in the field of war. Faster cell atrophy, that's been documented, and impaired long-term memory retrieval, and many other kinds of problems. But we actually know where this is happening in human animals, in us. So one of the questions is interventions. Given everything that you've heard already, what can we do? So there are many ways to work. One idea is stress management. So can you do the following? I have so much less stress since I removed the phone and the inbox, the computer, the email, the text messaging, etc. Well, we actually directly address this. There's such a thing as mindful email. We can or cannot talk about that later. But this, of course, is unrealistic. We have to live in the world, and we have to develop our abilities. So here's another key point. How do we shift from compulsion to choice? To what extent am I aware that I have a choice? To what extent do I not take the time to actually see what my impulsivity, compulsion, drives me towards? So one of the key ideas is how much does a person need to pause to be able to become aware more clearly of what are the compulsive behaviors, emotions, thinking patterns, what are the actual choices? And this, I would suggest, is directly trainable. In fact, everything that we're talking about here, even modifying cortisol, even modifying many levels of the immune system, the hormonal system, you can actually modulate it willfully. There's more and more evidence. It's, in one way, it's duh, of course. Why? Because this is all interconnected. So, resilience is one word that we might consider. The ability to recover quickly, skillfully, from an illness, from a change, from a misfortune. This can be important in terms of, obviously, in terms of relationships. It can be important in terms of just your email system changing at work increases cortisol in everybody, right? For the next several months, if not weeks, weeks or months. But on the other side, what does it mean, and this is a challenge, and this is more the provocative side, to develop equanimity, buoyancy, elasticity, psychological flexibility? And again, I'm going to drive this point that this is trainable. And does our culture, does our current educational system, mindfulness at schools, meditation at schools, actually support the cultivation of these qualities, which would last and have effects lifelong? My experience with Google engineers, computer scientists, MDs, PhDs, they're incredible high IQ, but when they show up to class, they're like, I have never been trained in any of these things that really matter. The highest level of leadership, greater emotional intelligence. Not part of anybody's education. Not even PhD psychologists. It's not part of our education. Big lacuna, missing point. So, but this is nothing new, and George loves this slide. One of our great, great heroes, 2 millennia ago, Marcus Aurelius, Greek Stoic philosopher. Wonderful book. If you haven't read it, get it. Meditations. And really, the point is that there's nothing new. If you are distressed by anything, external or internal, the pain, physical or emotional or spiritual, is not due to the thing itself, but to your own estimate of it. That's key. And this you have the power to revoke at any moment. This is called skillfulness, cognitive reappraisal, reframing, cognitive perspective taking. There's a lot of jargon and language and many, many more terms. But this is, the point is, here we are, 2012. Everything that we need already existed 2,000 years ago in terms of psychological health, skillfulness, mastery of the real research laboratory, which I always point out is our own body and mind. You don't need a grant to study it. In fact, this is the most important research laboratory. And the extent to which we miss that, we're suffering, and we're stressed, and we cause other people stress. That's even more important. We cause other people stress because of our own lack of doing research. Hence, when I give talks, I ask everyone, please be a scientist of this. So, if Marcus Aurelius put this out 2,000 years ago, how do we get it? Well, I work with James Gross at Stanford, psychologist, who is Mr. Emotion Regulation. So emotion regulation refers to many different types of skills or strategies. One simple definition is the alteration of ongoing emotional responses through the action of regulatory processes. Blah, 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 lots of words. The ability to willfully, automatically and trained, effortful and non-effortfully, implement strategies that begin to modify our emotions, intensity, duration, valence, amount of arousal, and to be an expression or suppression. So it's a whole set of skills, and this is really important because I think this is directly related to the ability to identify stressors, stress response or reactivity, and then to act on it. So there are many stages. So the red arrow here refers to something that happens. Somebody, say your partner, you come home tonight, your partner looks at you with a grimace or an angry or a contemptuous face. Within milliseconds, your amygdala is firing, your prefrontal cortex and other areas are going, oh my gosh, it's going to be a horrible night. And how do you begin to implement different emotion regulation skills? Where are the opportunities or the windows in which, from the moment that red arrow happens, an event, well, there are, as you can see, there's a whole range from attention regulation to cognitive thinking regulation strategies. So even on a pre-conscious level, we have evidence that if you train yourself in certain emotion regulation skills, they can begin to be implemented automatically, even before willful or volitional activation. That's exciting, but not happening in most of us. The next level is attention allocation. Where do I place my attention? How do I regulate my attention? We have beautiful research showing that there are clear neural circuits in which are instantiated different types of attention regulation abilities. But maybe I missed that opportunity. And then later is emotion appraisal. My partner is freaked out. So you appraise and you evaluate what is the emotion that's occurring. I'm scared. Boom. The next level, which requires more skill, and we already have all of these, but the whole idea is how do you refine it? Cognitive reappraisal, reframing, changing the meaning of a specific situation in order to reduce the toxicity. My partner had a bad day, she's upset, it'll be all right. I can handle this. Cognitive reappraisal, changing the meaning, it's not about me. using thinking, language, logic, different parts of the brain, mostly in the medial, dorsolateral, prefrontal cortex. Then there's another aspect, which is suppressing. Actively, willfully suppressing, showing your emotion. In some contexts, that can be very effective. If I show my fear, I might cause my 4-year-old to become more scared. So I will willfully suppress showing on my face what I'm feeling in this context for the benefit of the 4-year-old child. Long-term suppression of showing emotions, as was already pointed out in the discussion, is not, it leads to more mental and physical illnesses, as opposed to cognitive reappraisal, which is a more adaptive, health-promoting form of emotion regulation. And then finally, and more interestingly, more complex, but also more simple, both, are metacognitive strategies for working with your emotions and with stress-inducing stimuli. So there's a whole range of skills that one can work with. Very simple model of how this is happening in the brain. There's a stimulus. It can actually be a real threatening situation. You get an email that says, from your boss, it says, "I'd like you to show up in 5 minutes in my office." It might be completely neutral. It might be real or imagined, and we imbue it with the power to cause stress in us. Within milliseconds, thank goodness, human animals, us, have evolved through evolution to have our brains sculpted in a way to respond to things that are personally salient within milliseconds. So there's a whole limbic system that supports emotion reactivity, emotion detection, emotion generation, the amygdala, the insula, etc. And we all know that within milliseconds or seconds, our emotions can shift, our emotional state can shift to joy, bliss, happiness, fear, anxiety. This sends a bottom-up signal to recruit help from other parts of the brain, shown here in blue. Regulatory systems. Can I shift my attention? Can I apply my attention in a way that might modulate the ongoing emotional response? Can I engage cognitive regulation areas that change the meaning, or that give me cognitive perspective taking and see the situation from another point of view? When the blue, cognitive and attention regulatory systems, which are trained through education and experience, when they're functioning well, they exert top-down regulation to help modify ongoing emotion reactivity. When this system is working well, I suggest that this induces a sense of confidence, self-esteem, reassurance to handle any emotion, and I would go even further, that you actually enjoy all emotions and use it as a source of information without being afraid of feeling. When this system is not working well, hyper-emotional reactivity, deficient top-down emotion regulation, this is the basis for anxiety disorders, major depression, suicidality, stress, etc. But in human animals, there are 2 more bubbles that really make us different from other animals. The notion of self, how we represent, relate, use language, and view ourself, which is really clearly instantiated in brain systems that we can see with brain imaging, fMRI, and also language, the type of self-talk that we engage in. Now, as a clinical psychologist, this is, of course, apparent in psychotherapy, everyone's way of thinking, narrative, how they describe themselves. From a neuroscience perspective, there are clear circuits that show us when a person is engaged in language, cognitive regulation, conceptual self-thinking. The brain actually, in many cases, is much more clear than people's first-person report. In some cases, it's not. And that's a caveat. So this is one of the main tools that we use in our group, MRI, magnetic resonance imaging. It's a beautiful, beautiful tool. The math and the physics behind it are one of the most incredible expressions of the prefrontal cortex in humans. It's an incredible machine that allows us, it's like being able to get under the hood of a car and right into the engine. Getting beyond first-person speech, which has so many filters, letting the brain speak for itself and show us what it's doing. So, one slide primer of how do you get a functional magnetic resonance imaging dependent variable? Because most people don't know. So you're lying in the scanner, not always a comfortable place, but you're lying on your back, and I show you your own core negative self-beliefs about yourself. I'm not okay the way I am. Something is fundamentally wrong with me. I'm unlovable, et cetera, et cetera, et cetera. People's own negative self-beliefs. And of course, that gets neurons firing, circuits firing, and then those neurons are consuming fuel, glucose, oxygen. Sends a signal downstream to astrocytes saying, hey, open up the arteries, the arterioles, deliver more cerebral blood flow, blood volume, glucose, oxygenated hemoglobin to the sites where neurons are firing. That's the basis for what's called blood oxygen level dependent signal, which is the basis for much of the kind of functional brain imaging that we do. Lots of signal processing, and then you get these cute color maps, color-coded maps of activity, which you see in Newsweek and Time Magazine, blah, blah, blah. But our work begins at that point. Now we become functional neuroanatomical detectives. How does the brain activity, the pattern of brain activity across multiple regions relate to the experimental design, that relates to the psychology of the experiment, that relates to the healthy or clinical sample, that relates to the therapy or meditation training or aerobic exercise training? So it's a huge amount of bringing together multiple streams at multiple levels. And it's very challenging, but it's really fun. So this is, now you all know the dependent variable in fMRI. So attention. We always begin with attention because it's the gateway to all other higher order capacities. And I always think back here, in the back of my brain, as a little gateway or a door that allows signals to migrate to the prefrontal cortex where thinking, analysis, logic, perspective taking can happen. Has many components, multiple components. We'll speak about just a couple of them. It's a very limited capacity. It's happening in the brain. And here, I would suggest that the quality of our attention is the basis for prosocial behaviors, noticing, compassionate behavior. In fact, I'm working on empirical data going all the way from attention, emotion regulation, empathy, to compassion, and suggesting that the quality of attention is the basis for the glue of social interaction, empathy, and even compassionate behavior. So, Tong and Posner. Posner is the guru of attention, an amazing, amazing example of researchers spanning from basic to clinical to social, etc., educational. And the question here is, if you look at this green bar, cylindrical, the idea that they posed is there a quality of attention in each of us that is the basis for optimal performance? And how do I know when my mind is actually gravitating towards non-optimal, to a wandering mind? And there's empirical research showing that most of us spend most of our waking hours in wandering mind, which is associated with more negative emotions. On the other side, effortful training, where we're trying to harness and focus our attention in a very intense way, leading to mental fatigue. And the challenge is, do each of us, as you mentioned, George, individual differences is huge. Am I familiar with what it feels like when I'm in an optimally, attentionally balanced, dynamic state? And if I don't know what that is, I need to learn it, because that's where you have optimal performance if you are driving a car, a therapist paying attention to a client, reading a scientific article, meditating in a monastery, sitting right here paying attention. So this is essentially a challenge for us. How do you get there? Well, I would say that about a decade ago, until about a decade ago, no respectable scientist, maybe more, would stand up at an international conference and talk about contemplation, meditation, or even different ways of training specific aspects of attention through ancient practices. And again, nothing is new. These are already here. But Antoine Lutz is a fantastic researcher at the University of Wisconsin-Madison, works with Richie Davidson. He's done much of the seminal work in looking at the effects of different training programs in both healthy adults and trained masters, and really this is a very conservative journal, and that they put a statue of the Buddha on the front is really quite interesting. But there are many ways of training attention. And I'm actually going to guide you through that right now, because in a moment I'm going to show you the differential brain patterns that you literally are training like a muscle through these two different practices. So you can actually sit just as you are. You might want to just sit up. And this will be very short, but I want you in your own research lab right now to experience something so you can help interpret and understand the brain imaging results. So you can have your eyes open or closed, but I'd like, if your eyes are open, just bring your gaze down. Very simply, bring your attention to the sensations of your own breath, perhaps at the tip of the nose, inside the nostrils, simply harnessing and focusing with gentle intensity your attention to notice the subtle and gross sensations as you breathe in and as you breathe out. You might notice the coolness of the breath as you breathe in and the warmth of the breath as you breathe out. Notice the quality of your attention. Sustained, focused. If and when the mind becomes distracted, note it and bring it back to the sensation of your breath inside the nostrils. Now, I'd like you to shift to another form of attention training, which simply is like opening the lens of a camera, expanding the field of view, no specific object to focus on, simply noticing in the stream of experience from moment to moment to moment any thought, sound, image, sensation, memory, anything and everything that moves through your awareness, your attention, from moment to moment to moment. No agenda, nothing to push away, nothing to hold on to. There's no right or wrong at all. Simply noticing what is moving through your field of view, your mind stream. Okay, now please take a deep, deep breath. Open your eyes. Look back at me. How many people felt more distracted when you were doing the focused attention on the breath? By show of hands. A few. How many people felt more distracted when you're doing this open monitoring, open field of view? More people. Individual differences. And in fact, that varies throughout the day. These are just two specific forms of training, which we actually used in a couple of our studies. So not only science, but also journalists and even magazines like Time, Newsweek are now putting meditation practices on the front cover. And I would suggest that this is a direct response to the level and intensity of stressors that we experience. People are hungry for tools. And I would suggest that the word for meditation, bhavna, it means to cultivate, as you were just doing a moment ago, insight into the quality of your attention, and then even further, the processes of how this works. It's a tool for examining. That's essentially science, but intrapersonally. So, one program to work on stress that was developed 33 years ago by John Kabat-Zinn at UMass Medical Center, mindfulness-based stress reduction, a hodgepodge of practices to deal with chronic patients with chronic pain, physical and emotional, 35 years ago. And essentially what's happened now is that meta-analyses of this one program, and there are many kinds of programs, clearly demonstrate reductions in symptoms of stress, depression, anxiety, increases in indicators of well-being, social connectedness, quality of life, satisfaction with life. Just teaching some basic skills. So the program is a hodgepodge, and just as full exposure, we study many forms of psychotherapy, aerobic exercise, vipassana meditation, compassion practices, mindfulness. Mindfulness is only one kind of practice. There's formal meditation, sitting, guided. There's informal practices, taking pauses. And there's hatha yoga, which in and of itself is super powerful in changing physiology, helping to reduce stress, symptoms, etc. So that's why I use the word hodgepodge. John Kabat-Zinn here. One word, and some Buddhist scholars have actually suggested we don't use the word mindfulness anymore because it no longer means anything. The word itself, sati or smriti, Pali and Sanskrit, literally means to adhere attention. In some cases, it means to recollect or to remember, to stay in the present moment. And you'll see how John Kabat-Zinn has defined this. Paying attention in a particular way, on purpose, volitionally, in the present moment, not past or future tripping, but in the present. And the most difficult for Westerners, doing this in a non-judgmental, non-self-deprecating way. I'm really no good at this, I suck, I'm never going to be skillful, I can't train my mind, it's hopeless, give up. That's one way to think. That's the opposite of this. So, some brain imaging. Here, there are many ways to probe emotional reactivity, and we've tried many different ways, from shock, pictures, video clips, actors saying things to you, but one of the most powerful is your own negative, distorted self-beliefs. It's the most personally salient. And what you can see here, shown in the red circles, are greater neural response in a set of brain regions that have to do with conceptual self-view. And that totally makes sense. When you're spinning on your own negative thoughts, you're activating a conceptual self-referential circuit, shown here. What's nice is that when a person drops out of that conceptual self-circuit, we can see when they stop. That's what's so nice about having a brain system, because first-person report might not be fast enough or accurate enough, but the brain is speaking in milliseconds. Also, on the other side, in the black circle, is the amygdala and parahippocampal gyrus, part of the hippocampus. This is also super important because this is indicating that when you're spinning on your own negative self-beliefs, there's arousal, perhaps negative emotion, and even recollection. So this is how we set up the pattern of emotional reactivity. And then the next question is, well, what can we do to that? Well, as shown in the black circle, the right dorsal amygdala, one brain region, super important, has been pointed out in terms of fear, anger, emotion, arousal, activating the entire immune system, the entire body. How much can we actually modulate its pattern of activity over time through a specific training? So the mindfulness-based stress reduction training is only 2 months, every day, at least half an hour. That's very little. And the question is, if you look at the solid line, what you see is, when asked to react to your own negative self-beliefs, the black line takes a little time, but that's showing activity in the right dorsal amygdala increasing when you're spinning on your own negative beliefs, and then a cue comes on to shift your attention to your breath with your eyes open inside the MRI magnet. In the context of a powerful negative stimulus, shifting attention to an object, your breath, brings the amygdala down at baseline. Now you look at the dotted line, and that represents 3 months later, after training. And there are a couple of key points. The first point is that the red circle. There is a punctate increase in amygdala response to your own negative beliefs. This has been replicated in other labs, suggesting that one of the psychologically flexible shifts that occurs in the brain, mind, person, is you become more emotionally resonant. responsive. But the second red circle is also very important. You come back down to baseline faster. There's tons of evidence showing that when you use emotional stimuli with people with current or even past major depression, the amygdala goes on and it stays elevated, which is a clear indication of being stuck, ruminating, spinning, inability to attentionally disengage. One of the key things that we see with mindfulness and other forms of meditation training or other kinds of training is the ability to have greater psychological flexibility, but it doesn't mean numbing or avoidance, distraction, or lack of experience or emotional experience. So this is really promising, and this is a focused attention to the breath. So notice that. Now, clinically, why is this important? The red areas here that you see represent from pre to post 2 months of mindfulness training, increased ability to recruit sets of regions in the brain that have to do with visual engagement with an external stimulus. Most people with anxiety disorders, the number one form of emotion regulation is distraction or avoidance. In fact, there's empirical data now on just healthy adults. The one form of emotion regulation used more than anything else is avoidance. So I would suggest that most of us are avoiding most of the day. It's functional most of the time, except when you come home, you see your partner, and they need your full attention. And if you avoid in that moment, you're going to be in much more trouble. Hence, the red areas here suggesting that even in people diagnosed with anxiety disorders, they can mount greater attentional engagement even in the context of something that is toxic, their own negative self-beliefs, as opposed to turning off, shifting off. The amount that they can recruit brain activity is directly related to pre-post reduction in anxiety, a brain emotion anxiety relationship. That's now when it gets more exciting. Again, Michael Posner, Jin Fan have done fantastic work, again, standing on the shoulders of great heroes, delineating different aspects of attention and which neural circuits they're instantiated in the brain. The ability to alert or place your attention and be vigilant. The ability to shift attention, reorient as needed. And then the blue, top-down executive control. Okay, I get home, I see my partner's face, I want to pay attention. I want to implement attention regulation for my benefit, for her benefit. Top-down, goal-oriented regulation of attention. The brain is showing us. There are many other aspects of attention, but those are just 3. Whoops, sorry. Just showing that, in fact, from pre to post mindfulness training, 2 months, we actually see an increase in brain activity in areas that load on attention regulation ability. Again, just a little piece of evidence that attention regulation and the application or implementation is trainable. And you'll see why in a moment this is so important, and we actually do this at Google and now beyond. So here's the latest and coolest thing. This is related to the second form of practice you did, which was the open monitoring, or what's often called receptive awareness, where there's no specific object to focus on. You're simply noticing your experience from moment to moment to moment. That is a very different kind of practice than object-based. And so here is recent data, analysis I did just last week, brain imaging results showing that increases from pre to post mindfulness training for 2 months, increases in the top panel, which is the insula, and specifically the anterior part. The more you were able to recruit the anterior insula, which is an area that's very important in emotion, in awareness of visceral bodily experience or somatic processing, actually awareness of bodily sensations, the more that increased in patients with anxiety from pre to post was associated at 3 months follow-up, 3 months later, with decreased use of suppression. And I think as George mentioned or someone, suppression of emotional expression is related to much more negative emotional and physical profile. So interesting that an area that has to do with greater visceral awareness of what I am feeling in my body is related to less use of an automatic suppression, emotion regulation. That's really good. What you see below is the amygdala and the hippocampus. Again, amygdala, arousal, emotional awareness, directing attention. Hippocampus, if you go back to the definition of the word sati, mindfulness, it means to remember, to recollect, to adhere the mind to the present moment. Hippocampus does many, many things, but it's also involved with memory, encoding. And here, what we found is increases in activity in the amygdala and in the hippocampus from pre to post mindfulness training at 3 months later, follow-up, was related to increased use, both use of and self-efficacy, metacognitive attention, metacognitive ability, of cognitive reappraisal, a more adaptive form of emotion regulation that has long-lasting beneficial effects. Different brain regions changing pre-post training related to decrease in maladaptive suppression, increase in adaptive cognitive reappraisal in patients who normally do just the opposite. They suppress and distract. They are not skillful in thinking or reframing. So what I'm going to show you is super important. So we started with how do you get from the state of stress, anxiety, angst, insecurity on the left to resilience, psychological flexibility, innovativeness. I would suggest that the effects of different meditation practices is WD-40. Is there anybody here who does not know what WD-40 is? Okay, simply put, right? The grit, the dirt, the automatic thinking, emotional, even biological patterns that we have developed over time become sticky. They block the ability for the key to go into the lock. WD-40 loosens up the grit, these automatic, mostly not conscious, behavioral, emotional, even biological patterns. So that is my metaphor for loosening up even the neural circuits that allows any form of training to become more effective. In fact, there's been some suggestion that doing mindfulness meditation before you go into cognitive behavioral therapy for depression, anxiety, eating disorders, et cetera, might enhance the skills that are learned in cognitive therapy, which are in some way easy, but in some ways very difficult to do. So that's one key thing. But now here comes the next thing. So what? Everything I've said so far, so what? Cute, scientist, nice, fun, brain imaging, colors. So what? The social impact, which was the third word in the phrase of this conference. So what? What's the actual relationship to social impact? Practical application. Here it is. Ready? There are many ways. Here's one way I'm involved with. My second job is Google. So 4 years ago, I was brought in to develop mindfulness-based emotional intelligence leadership training for Googlers, for the reasons that I mentioned earlier. Huge stress, emotional reactivity, and lack of education in emotion regulation, and clear evidence, both within Google and outside, that the people who are the top managers, leaders, VPs, are people who manifest emotional intelligence skills. And it was not part of anybody's education. So when the Googlers come to the class, they're hungry because they know this is the only way to really, really advance unless they're going to be an individual contributor and not work with anybody, which is allowable at Google, not other places. So this is the book that's coming out April 24th that actually has most of the content. We are making the entire program open source, freeware, with everything. Books, slides, teaching, boom, 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 because the explicit goal is to actually enhance people's ability, optimize their performance, their well-being, not only in the workplace, but any organization. And even beyond that, I would actually say the goal is world peace, because this has already been taught all over the world in Google offices. And the lawyers just gave us permission to take it outside of Google. Thank you, lawyers. So that's coming. Lots of people help me. I have my mentors. You always thank your mentors at Rutgers, San Diego, Stanford. Thank you for your attention and funding. And let's go back to thank you for your attention.
Thank you very much, Philippe. We don't have much time for discussion, but we're going to have plenty of time at the recapping at the end. So I will invite our next speaker. Our next speaker is Professor Pathik Wadhwa from the University of California at Irvine. His lecture is on early life stress, infancy and childhood, and its impact on society. Pathik.
Great. Thank you, George, for the invitation and for this opportunity. So I'm going to try to follow the talks of this morning, particularly the talk by Dr. Chrousos and then by Dr. Romero, and speak to the issue of some of the effects of in utero stress exposure on long-term health and disease risk outcomes. And this talk really represents an effort by some of my collaborators and me, and so I want to acknowledge Claudia, who's here in the audience, and another of our collaborators in putting this together. We run a program at UC Irvine called the Development, Health and Disease Research Program, and in a nutshell, the goal of this program is to study the interplay between biological, behavioral, and psychosocial processes in early life. And when I say Development, Health and Disease Research Program, what am I referring to when I use the term health? So I just put this up to tell you that these conditions represent the major burden of disease in society today. So you're familiar with these conditions in adult life. It's heart disease, type 2 diabetes, cancers. In early life, it's premature birth, childhood obesity, asthma, neurodevelopmental disorders. For the first time last year in human history, these non-communicable disorders represented the major global burden of disease in the world. So this actually last year changed positions with infectious diseases, according to a WHO report. So for these particular conditions, these are all complex common disorders, common because they're common, complex because they have a multifactorial etiology. And if you ask the question generically, what represents the likelihood of an individual developing one of these complex common disorders, it would probably be a joint function of exposure to risk factors for that specific condition. But we also know that a group of individuals exposed to the same amount of risk, the differences, individual differences in who develops a disorder and who doesn't, and that's subsumed by the term susceptibility or vulnerability. So it's a joint function of exposure to risk and the individual's vulnerability. The traditional model has been that this vulnerability or predisposition has been equated to genetic makeup. So they say there's a genetic susceptibility and then there's exposure to risk factors. And the newer thinking now is that yes, genetic makeup matters, but it's really a combination of the genetic makeup and the environment in early life that together defines the structural and functional integrity of systems. So in a nutshell, you have something that works. It's exposed to wear and tear over time, depending on the conditions in which it's operating. And so one factor that goes into disorder is the amount of wear and tear that a system's exposed to. And the other factor is how well, how robustly, how strongly that system was built in the first place. And what I'm saying is that the building or construction of a living system in the first place is not merely a function of the genetic makeup at the time of conception, but it's the interplay of those genes with early environment, the emphasis being on it early. So So in this presentation, I'm hoping to spend some time talking about why one would consider a role for maternal stress and stress biology in the context of long-term implications for health and disease risk. Then share with you some summaries of some of the work that we've been doing related to this question. Move to discussing some what we believe are novel approaches for the characterization of psychobiological stress in pregnancy, and then end by briefly showing you an overview of the current studies. So if one didn't have any empirical data, would one even propose or hypothesize a role for stress in early development? And I would submit to you that one would, and this is based on concepts in evolutionary biology, developmental plasticity, and life history theory. So essentially, at the end of the day, from the same genotype, from the same fertilized egg, one can get many different phenotypes. You know this, animal breeders know this well, botanists know this well when they take the same seeds and the different conditions, they're grown at different altitudes and so on. The same is true with mammals, including humans. So what is it about this journey from genotype to phenotype that determines what specific phenotype a particular genetic makeup is going to acquire as opposed to a different one? It's really the process of the interplay with conditions during development or environment during development that defines this. And I'm using the term environment or conditions during development, and you might say, well, which conditions? That's a broad term. What are you talking about? And I would submit to you that if one considers what the key environmental considerations are that have driven evolution and natural selection, then one could reasonably expect that those same conditions would probably influence the development of an individual in early life, in intrauterine and early postnatal life. So what are those conditions? The one absolute obligatory factor that all life needs from the outside is energy, because no life on this planet is capable of de novo synthesis of energy. So energy capture, nutrition, getting energy substrate, anything that deals with how much there is and the efficiency of capture would be a key process that drives not only evolution but lifespan development. And the second is, once one gets energy, how do you regulate that energy? Under what conditions do you store how much, use how much, what place, what organ systems do you supply more to, less to? This whole thing is, at the end of the day, driven by the stress response. That's the fundamental reason for the stress response, is reallocation of energy depending on circumstances. And also, stress response obviously comes into play when there are threats, either actual or perceived, to either survival or reproductive fitness. So for these reasons, stress would be something, at least biologically, that developing organism considers very carefully in terms of the specifics of how it's going to develop. So again, two reasons. One, because it represents threat for mortality and reproductive fitness. Two, because the stress response is fundamentally the response that regulates the allocation of energy in terms of efficient capture and where and how much to store and use and keep changing this. I'd submit that's the key. There's another reason why one would consider the effects of a stress, which has to do with the very large socioeconomic and racial ethnic disparities in all the common complex disorders I showed you, starting with reproductive and birth outcomes and moving on to the other conditions. And we know that health-related behaviors only explain a proportion of these socioeconomic and racial ethnic disparities, and there's been a hypothesis for a long time that differential exposure to stress may be playing some role in explaining these disparities. So that's another reason why one might consider a role for prenatal stress. So in summary, I'm suggesting to you that stress represents cues or signals about certain key environmental conditions in the context of development. The stress system represents probably a very important communication system between the maternal and placental compartments. So the same stress mediators that represent cues also communicate this information. And then these same stress mediators, glucocorticoids, cytokines, actually act on various targets of programming. And so it's a very efficient way of collecting, transmitting information, and then producing effects based on this. And we're going to get into a little bit of this. So it's for all these reasons that, at least for me, the study of stress and stress processes in early life is very interesting. So moving to a little bit of a summary of some of the work we've been doing. We started several years ago with this question: does maternal stress during pregnancy influence fetal development and subsequent outcomes? We asked several specific questions: which aspects of stress, which outcomes does it influence? Is this really a stress effect, or is this an epiphenomenon? Is it because these women are experiencing obstetric complications and are worried about that? If there is an effect, what's the magnitude and nature of the effect? If there are effects, are there critical periods in gestation when pregnancies and embryos and fetuses are more susceptible than other periods? What are some of the mechanisms and how are these signals transduced? So, excuse me. So essentially, we started with studies of psychosocial stress exposure in pregnancy, and we started by considering the role of the endocrine system. But these studies have evolved over time, and we now, in addition to psychosocial stress, are interested in the effects on nutrition and are doing studies on that, in the effects of infection and the interplay of the three: psychosocial stress, infection, and nutrition. From the endocrine work, we quickly moved to also including the immune system and to studying some genetic and epigenetic processes. And from pregnancy and birth outcomes, such as premature birth, now we move to some of the longer-term effects. I'll show you some of that. If one looks at the role of maternal psychosocial stress in adverse pregnancy outcomes, such as preterm birth or small for gestational age, if you summarize the effects across studies, you see what one might describe as modest effects. So the pooled averages, most of the studies do suggest that there is an effect such that women reporting higher levels of stress during pregnancy tend to give birth sooner and give birth to smaller babies. This effect is pretty modest. So the two questions: one, there's also, in addition to the modest effect, there's a tremendous amount of heterogeneity in these studies in terms of the study design, the conceptualization and measurement of stress, of outcomes themselves, and so on. So the two possibilities here: one, that the effect really is modest, there is a real effect, but it's nothing to get too worked up about, or that we are not perhaps measuring this in terms of whether there are some specific individuals under specific conditions other than stress that go with stress at specific stages in gestation who might be particularly vulnerable to these detrimental effects. So I'm going to come back to this issue a little bit later and talk about how we're trying to address and study this. In terms of biological pathways, these are some of the common pathways: endocrine, immune, inflammatory, and vascular, that are discussed in the context of the mediation of stress effects on fetal development. And I'll say a couple of words about each of these. So two things that are interesting about endocrine processes in mammalian and particularly in primate pregnancy are that primates, humans and non-human primates, are the only mammals that in the state of pregnancy produce large amounts of CRH in the placenta. You've heard a bit about CRH from Dr. Chrousos, and this is the hormone that organizes and mediates the endocrine stress response. Now, this is made by the placenta. This hormone is quote-unquote stress sensitive in that its production is regulated by all the known mediators of stress such as cortisol, catecholamines, neuropeptide Y, inflammatory cytokines. And it's known to produce important effects on two sets of processes. One is in regulating the length of gestation, determining the timing of birth, and the other is regulating various aspects of growth and maturation. It's the combination of these two, the synchronization of these that is important in mammalian development. So we've published several studies that suggest that maternal psychosocial stress is associated with dysregulation of the maternal HP axis, which in turn correlates with placental CRH production, which in turn correlates with birth outcomes, such as preterm birth and fetal growth restriction. The next set of studies, we began examining the interface between stress and infection in pregnancy. You heard a little bit from Dr. Romero about the role of intrauterine infection in pregnancy. And we have been pursuing two hypotheses and have published a little bit on each of these two questions. One, the idea that maternal stress, women reporting high levels of stress are more susceptible or vulnerable to developing reproductive tract infections. And the second is that among women with established reproductive tract or intrauterine infections, those reporting higher levels of stress seem to have, it seems to exacerbate the pathophysiological consequences. So in fact, we've shown, and Dr. Chrousos mentioned this, CRH is a potent agonist of inflammation when antigens are presented. So in the non-infectious state, we used maternal blood in early gestation. There was no association of CRH levels with cytokines, but when we stimulated the blood with LPS, you saw a robust pro-inflammatory response, individual variation, and this variation in the pro-inflammatory response was very nicely explained by levels of CRH, such that women with higher CRH had a more robust antigen-stimulated response ex vivo. We did this with both whole blood and with PBMCs. We're also pursuing the role of stress-related gene-environment interactions in pregnancy, and we're considering several stress-related, HPA-related genes in both the maternal and fetal compartment, and the approach is to look at various combinations of maternal as well as fetal genotypes, always in conjunction with environmental exposures such as stress or other exposures of interest. And so that's some of, that's just a sort of brief summary of some of the work looking at adverse birth outcomes. Moving to some of the longer effects, we're interested in exploring the long-term effects in the brain and in the periphery of intrauterine conditions with a focus on stress. So you've already been introduced a little bit by Dr. Chrousos and then more by Dr. Romero to this concept of the developmental or fetal origins of health and disease. If one had to summarize this in one slide, it would probably be this, that many epidemiologic studies all over the world have now established a correlation between markers of birth phenotypes, such as birth weight, and risk for disease in later life, such as hypertension, coronary artery disease, diabetes, and so on. The assumption here is that these adverse birth phenotypes are markers of adverse intrauterine conditions. So we've, among the questions we're interested in is this question: are the effects of prenatal stress exposure necessarily mediated by adverse birth outcomes, or can you get subtle effects that you can pick up later in life despite having what appear to be normal pregnancies and normal birth outcomes. So we again, are looking at brain development and periphery, and one of my collaborators who is focusing on the effects on brain development is here, Dr. Claudia Buss. She's published several studies. She started her work looking at adverse birth outcomes, again, as markers of adverse intrauterine conditions, and looked at those and the interplay of those with adverse postnatal conditions on several measures of structural and functional outcomes in brains, in starting with adults and then moving into children. And now this work has become prospective, and I'll give you a couple of examples. So you heard again this morning about the amygdala, the importance of the amygdala in the regulation of emotions. You heard some references to the circuits in the amygdala, and this, I believe, is one of the first set of data. It's unpublished as of now, but we believe in a couple of weeks it's going to be published in PNAS. This is one of the first pieces of evidence that links in utero stress exposure, in this case glucocorticoids, with the size of the amygdala in children, and in turn links that with emotion regulation and affective processing. So it sort of completes this structure-function relationship and ties it, correlates this with prenatal stress exposure. So as I said, in about 2 weeks, we should be seeing this paper coming out. Here's another finding that Claudia has developed that speaks to the importance not just of prenatal processes, but even characteristics that a mother brings to her pregnancy. And you've heard this morning about obesity and the inflammatory milieu that accompanies obesity. And in this finding that Claudia has developed, she links pre-pregnancy BMI with the cortical thickness in children as analyzed with brain imaging. So after controlling for child age, child BMI, birth outcomes, and so on, she found that cortical areas, particularly in the prefrontal cortex, are reduced in women with a higher pre-pregnancy BMI, controlling for weight gain in pregnancy. And we think that this potentially is tied to a risk for higher impulsivity, less inhibitory control, which we heard discussions about in terms of the implications for psychopathology, for ADHD, and possibly even for obesity risk later in life. Our other colleague, Sonia Entringer, has been asking this question in terms of physical health phenotypes, endocrine, immune, metabolic. Is there a direct relationship between measures of exposure to prenatal psychosocial stress and some of these phenotypes that's independent of birth outcomes? And so the first study she did examined this using a retrospective case-control design. You heard again this morning about some lists that represent very severe stressors that most people would, we all know that there's a lot of variation in the middle range of what we'd consider stressful, what I might consider stressful, he might find not only not stressful, but enjoyable and challenging. But there are some extreme conditions that most of us would probably agree on, and we saw a list of those, like the death of a spouse and the loss of a job and a severe illness of somebody. So these were the sorts of stressors that Sonia looked for in terms of a history of prenatal exposure. She decided to focus in this study on young adults, healthy young adults, where there was no evidence right now of disease, because while psychological state can produce disease, disease itself alters psychological state. So she didn't want that confounded in this study. She made sure from obtaining the actual medical record in the index pregnancy that there were no major obstetric risk conditions, no adverse birth outcomes in this study. She looked at a whole bunch of metabolic, immune, endocrine, cognitive outcomes that I'll discuss in a minute. And she used a lot of challenge tests to look at these. So these are healthy 25-year-old men and women. Half of them have a history of exposure to prenatal stress in the index pregnancy, where the mothers experienced, when they were pregnant with this person who's now 25 years old, the death of someone in the family or the loss of a job or a severe illness. So you follow the design. Except for this, nothing untoward in the pregnancy, no adverse outcomes. These 25-year-old men and women are perfectly healthy on all measures right now and have been healthy through childhood. So we brought them into the lab and performed various assessments, physiological assessments. She looked at challenge tests because if you are interested in knowing whether I'm at risk for, let's say, developing hypertension, and if you measure my blood pressure at rest, it's not as informative as if you make me exercise and measure my blood pressure in response to a challenge. If you're interested in knowing whether I'm at risk for developing diabetes and you measure my blood glucose at rest, it's not as informative as if you were to give me a glucose load and then look at how I regulate blood sugar. So when you challenge a system and you watch how the system responds to a challenge, that's when you first see signs of dysregulation, and you see those well before, years and years, sometimes decades before overt disease sets in. So these were the sorts of tests she used in looking at endocrine, immune, metabolic, and cognitive functioning. I won't go into the details of those, but I'll walk through quickly some of the results. So she found that the offspring of mothers who'd experienced a high level of prenatal stress had a higher BMI, were already by 25 years age insulin resistant, although they were not diabetic, and they had a lipid profile that's consistent with the metabolic syndrome, low HDL, high VLDL. In terms of the endocrine system, they were producing, in response to a challenge, more ACTH and less cortisol. This is something that has been noted in the HPA axis of people who are particularly prone for psychosomatic disorders. She got blood samples from these young men and women before and after stress and noted a shift, a Th2 shift in the Th1-Th2 ratio, suggesting an immune dysregulation that places these folks at risk for allergic and atopic disorders. In terms of memory, found memory impairments under stress. So she looked at memory both at basal condition and then once again after administering cortisol, and found that the post-cortisol memory, or the memory under challenge, was impaired. And then finally looked at a marker of cellular aging, which I'll discuss a little bit more, telomere length. So we tend to think of fetal life and childhood and young adulthood, and then we think separately of aging. And we ask the question here, does the rate of aging, cellular aging, does that have anything to do with conditions in intrauterine life, and does stress play a role in determining how one ages? So for this, we were interested in, and we've recently published this paper, and I know Dr. Chrousos was one of the reviewers. You've heard some mention about telomere biology, and just to remind you, these are the ends of, these are DNA sequences that are at the ends of chromosomes, and they have a loop. They protect chromosomes from damage to the DNA base pair regions. They get shorter every time a cell replicates, but they get elongated when an enzyme called telomerase is expressed. So the ultimate shortening is the shortening with replication and the little bit of elongation with telomerase when it's expressed. So in balance, there's some shortening. There's some recent studies. There are many conditions, and Dr. Chrousos showed you some of those, that telomere lengths associated with people with shorter telomeres die sooner, more likely to have heart disease, diabetes, a whole bunch of other conditions. In addition to these now, in the last year, there have been some, what I consider landmark papers that suggest that telomeres and telomere biology fundamentally regulates all processes to do with aging in cells. And it does this by maintaining the tightness with which DNA strands are wound together. So one hallmark of an older cell is that the DNA is not as tightly configured. And this is because histone production gets compromised in old age. These studies now show that histone production, through some signals that are not yet identified but that emerge from telomeric regions, regulate histone production that's responsible for global second-order chromatin integrity. And so experiments have demonstrated some of this, have demonstrated in terms of senescent cells that if you just remove senescent cells, which again is a function of telomere length and telomerase, you get a dramatic, if not reversal, at least an arrest of the aging process. So we looked at telomere length in these young men and women, and lo and behold found that those who'd been subjected to prenatal stress had shorter telomeres. The shortening was about the equivalent of 3.5 additional years of aging. So already by the age of 25, the cells look 3.5 years older in those who'd been exposed to prenatal stress. So now moving to some of the ongoing work, remember I said to you that there are two possibilities. One, that the effects of psychosocial stress on adverse birth outcomes are either modest, or there's some women or fetuses under certain conditions at particular stages in gestation who might be particularly vulnerable. So if one asks the question, well, what is the likelihood of a stress-related adverse health outcome? Again, using the same logic, it would be a function of two things. One, the amount of exposure to stress. But the second would be individual differences in the biological responsivity to stress. So if, for instance, if the fire alarm went off, you saw some smoke coming into this room, if we all believed that there's a fire, all of us would experience some degree of stress, mentally and physiologically. But then, a few seconds go by and someone comes in and says that was a false alarm, everything's okay. We'd all settle down and get okay. But there'd be quite a bit of variation in this room about who remains agitated for how long and who gets calm quickly, and whose blood pressure and catecholamines and cortisol come down quickly versus stay. That's the psychobiological stress responsivity component. This is something that hasn't been looked at in studies of pregnancy in terms of the response. So what we're doing now is, in the most recent studies we're doing, we're working very hard, and luckily we have the cooperation of our research subjects. We're instrumenting them with gadgets such as what you see in this picture, that collect 24 hours a day, that collect EKG, respiration. With an accelerometer, they collect physical activity and posture. The combination of these gives us sleep. And then they complete an electronic diary about 15 times a day. So we do this thing 4 days in each of the 3 trimesters of pregnancy. So they spend 6 days with us, 1 day to train and 1 day to come back and return the equipment. And they collect these data for us for 4 whole days. The only time they don't collect these are when they're taking a shower. But otherwise during sleep and during natural living conditions, we get quite a bit of physiology. From the diaries, 15 times a day, they tell us where they are, what they're doing, how they're feeling, what they've eaten and drunk recently. We also collect 8 samples of cortisol a day. So I'm not going to go into the details of how we combine all this data, but I'll tell you that we get what we believe are far better self-report data about how they're doing than the usual questionnaire interviews that have you ask me to think back over the past week of my life and summarize on average how I've been feeling. This is in real time. And then you get data about physiological state. You can combine these, you can create averages for what average stress levels are, and then combine these to look at psychobiological stress reactivity. And this approach looks like it's working. We've published some preliminary studies on this with the first few subjects. We now have projects that are doing this protocol and then following up the babies with, in terms of our interest in brain imaging and in understanding metabolic function. So instead of focusing on discrete conditions like psychosocial stress or infection or obstetric complications, we're focusing on looking at an array of biological mediators of stress, such as cortisol, CRH, IL-6, C-reactive protein, because each of these biological stress mediators is driven by any of the antecedents. And again, we've heard Dr. Chrousos say a little bit about some of this. So we're measuring these longitudinally over the course of pregnancy, all the way till birth. These are our markers of prenatal stress exposure, and we'll be characterizing the exposure specific to just stages of gestation, cumulative. We'll be looking at trends of change over time as well. And then in one of the projects that Claudia is heading up, she's looking at the vulnerability hypothesis, which says that vulnerability for neurodevelopmental disorders is in part a function of alterations in the brain, which are produced in part by the influence of prenatal conditions. So she, in this study, is doing very extensive brain imaging with MRIs, with diffusion tensor imaging that looks at fiber tracts. Also now she's using resting state fMRI. You've heard some talk about fMRIs, and these are resting state, these aren't challenged. Looking at these in infants during natural sleep, looking at several postnatal conditions as modifiers. And I've already shown you some examples of the data she's produced. Another of our colleagues is looking at susceptibility for obesity. So no matter what, one cannot become obese if one doesn't consume more energy than one expends. That's the bottom line. You just cannot become obese. But among people who consume the same, more or less the same amount of energy for body weight, there's huge variation in who puts on weight and who doesn't. This susceptibility is something that we believe may have something to do with developmental conditions in intrauterine life. So it's not actually obesity, but it's the susceptibility for that under equivalent conditions of energy balance. And that's what we're looking at in this other project, where we're doing, using DEXA scans to do whole body imaging, using blood samples to get at insulin sensitivity, and then doing measures of energy expenditure using a procedure called the doubly labeled water method. Also focusing on the role of postnatal nutrition, breastfeeding, and the constituents of breastfeeding in this project. So, to wrap up, if one talks about prevention of stress-related disorders, I think it's reasonable to suggest that primary prevention would begin in intrauterine life. If one starts thinking about doing something about stress, even after birth, it may not capture the whole spectrum of susceptibility that stress produces. I read this quote somewhere and I like it, and I think it's apt here. The wealth of a society lies primarily in the health and well-being of its girls and women of reproductive age. And that certainly couldn't be more true in terms of susceptibility, both for males and females in life. And I at least hope I've suggested to you that stress biology is interesting in its own right in terms of the effects of stress, but it's far more than that. It has the potential of offering a unifying framework in the context of framing questions about mammalian adaptations to environmental variation, because it is the system that's used to detect, to transmit information and the system that produces effects based on this. So I think it's a very good model system in understanding the effects of environmental variation. So all of us have different ways of dealing with stress, and these are my personal stress buffers. The dog's more than my wife, but we won't tell her that. And I have the fortune of collaborating with many, many wonderful people at several institutions. All of this work is funded very generously by grants from the NIH, so thank you.
I think we may have time for one or two questions. Dr. Simopoulos.
That was an excellent presentation.
Thank you.
I wonder, in the data that you are collecting, whether you have data on fatty acid intake in terms of arachidonic acid, EPA, and DHA in the mother, at the cord blood, and then during the first 2 years of infancy?
So I don't know exactly. I'll tell you what we're collecting, and maybe the information's there. So number one, we're using this NR software to get at 3 24-hour diet recalls in each trimester. Are you familiar with that methodology that asks people to list in great detail? So if these constituents can be picked up there, we'll get that. That's point one. Then the second thing is we're collecting plenty of maternal biological samples in each trimester. So we have, starting with maternal hair, maternal saliva, plenty of maternal blood, and maternal cervicovaginal fluid. So not in the cervicovaginal, but at least in some of the other analytes, but blood. We have plans for looking at some nutritional biomarkers. We're not funded to do that, but we have an interest in that. We do not, at the present time, have funding to, in this new set of studies, to collect cord blood. But we've just applied for a new grant on a supplemental project. So if we get that, then we will have cord blood, but at the present time we don't. And then last, in early postnatal life, we're again administering nutrition questionnaires, but also collecting breast milk samples, both in the early, mid, and late part of a typical feed.
Yeah, well, you actually need either plasma or red cell membrane phospholipid levels to know exactly how much is getting across the placenta to the baby from the cord blood, and later on, because there are genetic variants in the pathways of the omega-6 and omega-3 fatty acids, so that a dietary history is not adequate. One, and secondly, in terms of epigenetics, both the arachidonic acid, NEPA, and DHA, they have enormous influence on DNA methylation and histone modification. And in terms of development and cancer, this area is expanding by leaps and bounds. And in fact, it becomes terribly important in terms of IQ. CASP has found out that certain genetic variants make a difference in the IQ.
Sure.
So I think it's important to know the genetic variation and the epigenetic variation, which is much larger than the genetic variation.
So we, of course, have DNA from the kids and from the parents. The epigenetic, it's limited as to what we can study, but it's just, maybe it's my personal bias, but I think the majority of studies associating epigenetic changes with exposures, in this case in early life, are suspect because most of the epigenetic changes, or a large proportion of them, are not stable. For instance, we heard this morning from Dr. Chrousos that maybe you didn't show the slide, but you have a beautiful slide that shows how circadian variation changes about 15-20% of the expressed genome. Some of these effects are through glucocorticoids. So depending on what time of day one looks, there'd be changes. These are all 100% epigenetic. And it's not as stable as it's being sold right now to people, that this is a process that is stable and stays in this state forever. Most of it doesn't, except the epigenetic processes that actually differentiate cells from undifferentiated into, liver, pancreas, muscle. A lot of the other stuff is not stable. So I don't question the importance of studying and understanding epigenetic processes, because that's one of two ways by which you regulate gene expression. No question about the importance of that. I don't think that a lot of the studies right now that take one-time samples and show these differences between state A and state B will replicate.
You could study the methylome, which is relatively stable.
Even that...
acetylations, etc., are not stable and change all the time, but methylations, you
no,
may want to
so I thought that till a week ago. Where was that? We found a paper, what was it on folate? I'm trying to remember the paper that looked at methylation, and showed surprising degree of change. I would have argued exactly what you said. We recently looked at children till a week ago.
With obesity, and there are a lot of permanent changes in the methylome of these children, which may explain why they don't respond very well to any treatment.
But I agree with you on the importance of this. I don't know.
Genetics and nutrigenomics that focuses on that.
Yeah. I think you have the samples, and you could do great things with these samples. Just keep them at -80.
We could. We certainly are storing the samples, yes, no question about it.
Any other questions? Thank you very much, Patrick. It was wonderful. Our next speaker is Dr. Laura Dorn from the University of Pittsburgh, right? Cincinnati. Still my jet lag is not functioning. Dr. Dorn will talk about stress in puberty and adolescence.
Okay. First of all, thank you very much to both Dr. Chrousos and Gold for the invitation. This is great. I think the conference has been really good. And everyone so far has set the stage for the next talk. So, George, I think you had the hardest job because you were first. So, okay. I have 5 primary goals today that I'm going to do. First of all, I'm going to talk about biological transitions as a time of vulnerability. Second, adolescence and puberty. And then specifically, I'll talk about changes in puberty, both normal changes as well as early changes. Third, the health paradox of adolescence. And fourth, I'm going to give some examples of the impact of stress on various outcomes in childhood and adolescence. And then finally, I'll talk a little bit about opportunities for intervention for this. Okay, what are biological transitions? Well, I had the good fortune of working with George on this paper several years ago, where we talked about biological transitions are times of vulnerability, of increased physical or psychological vulnerability for some. Now, we've already heard some of that today with just the most recent presentation. This vulnerability may be related to changes in reactivity of the stress system. It may be genetic or constitutional, and the context may modulate the effect of the transition. So many of those terms we've heard already today. Biological transitions are processes that primarily occur during reproductive phases across the lifespan. We heard about pregnancy this morning, for example. They involve distinct hormonal changes, and they may or may not be accompanied by physical changes that are external as well. Here's puberty. This is a lot of people's vision of what puberty is. It's a big time of change very stressful for some. But the next slide actually shows you what the adolescent perspective of puberty is. This was on a t-shirt I saw, actually. Puberty is when parents are difficult. So maybe we're actually studying the wrong thing. We should be studying parents during puberty. Okay, I want you to keep adolescence and puberty as kind of two separate things. Adolescence occurs basically in the second decade of life with psychosocial changes. Puberty is a part of adolescence. It occurs during adolescence. More recently, we found out that there's an increase in stress-mediating activity during puberty, and that this increase probably partially mediates the risk of emotional pathology in adolescence. Okay, I mentioned there are multiple changes that occur during adolescence, including puberty. I'm going to talk a little bit about puberty now. First, that includes, as you know, sexual maturation and also physical growth. Then during adolescence, you see cognitive developmental changes, changes in motivation and emotion, maturation of judgment and self-regulation skills, and then brain changes are actually linked to each of those components. And then, of course, during adolescence, there are various social and role changes that we're all familiar with. In normal pubertal development, there are really two components that actually have some overlap. The first component people don't hear about very much. It's called adrenarche. It means awakening of the adrenal glands. You get higher concentrations of about 3 different adrenal androgens. Those hormones, at least during puberty, tend to be responsible for things like development of body hair and pubic hair. Gonadarche is the next component that we've all heard about. Nowadays, it happens as early as 7 or as late as 13 for the initiation. And you get the rise, a distinct rise in several gonadal hormones during that time. We think it's earlier in girls, and I'm going to show you a few slides about that because that has something to do with a lot of the stress research that has been going on, at least in adolescence. This slide on the left depicts sort of normative order of changes that occur. There could be a lot of variation here. If you look at the left, which is for girls, you can see that breast budding, which usually is the first change of puberty, can be as early as 8-ish or as late as 13-ish for the initiation. And then full adult breast development might occur as early as 11 or so, all the way up to 16 or 17. Huge variation. So if you're that girl, it makes a big difference where you fit in terms of timing. If you're the parent of that girl, it also makes a very big difference. You can see the first period is could be anywhere from 9, 10 or so, maybe not until 15 or 16. And I want you to notice that that is not the initiation of puberty. That's about a mid-process change because puberty is a process. For boys on the right-hand side, you can see that usually the first change for boys is change in testicular size, the first bar at the top there, at about 10.5, could be that early, all the way up to 17 for completion. And peak height velocity, you can see, is different for boys and girls. And boys tend to mature physically about 1.5 to 2 years after girls do. So is puberty in girls beginning earlier than previous years? I think the answer is probably yes. For a number of years, we weren't sure, but now I'm fairly convinced that that's true. The age at onset is now about age 7 to 8 in some girls, and I say some girls because it's not in all girls. And the age at menarche has dropped across the last decades or even century. This study was published in 1997 and actually had a lot of controversy with it. Herman Giddens did this study with over 17,000 girls, And you can see here, it's talking about development at Tanner 2 or above. Tanner 2 is the first stage where you enter into puberty. So in this study, at age 7, in the dark bar, African American girls, about 25% were already at Tanner 2 by age 7, compared to about 5 or 6% of Euro-American girls. By 8, those percentages really changed dramatically. Now, there have been several other studies since this time that corroborate these findings. Early in puberty in girls, I mentioned, could be a stress, and there really is quite a vast amount of literature that does support this in terms of girls being more at risk for both physical and mental health sorts of issues. Girls can have shorter adult stature if they're early matures. There's also some indication for higher rates of breast cancer as an adult, depression, substance use, behavior problems, et cetera, et cetera. There's many, many studies on this. There's less literature on boys in puberty. Some studies do show that boys with early maturation may be more likely to have higher rates of testicular cancer. Now, girls with one type of early puberty called premature adrenarche, remember that's awakening of the adrenal glands. These girls are at risk for physical disorders in adulthood that I've listed there. Now, those probably look familiar to you. Dr. Vadva just described some of the studies with girls with early birth, small for gestational age and perhaps prematurity, may be more at risk for some of these. Anecdotally, it seems like a lot of parents with girls with premature adrenarche mention about all the behavior problems and that come with that. So we actually did a couple of studies to focus on that, to see are they at risk for those sorts of issues. This study was published in 2008, and it had 40 premature adrenarche girls in the light bars and 36 healthy comparisons. And what this is showing along the horizontal axis is a behavior problem checklist score that parents rate their children on. We took and figured out who hit a 60 score, which was considered clinically significant. And to you and I, that if you were the parent of that girl, you would notice this behavior. If you were the teacher, you would notice this behavior. And the PA girls had nearly 25% of these girls had hit above the 60 mark for social problems and total problems on the far right, and also aggressive behavior compared to the healthy comparisons. Now, I also want you to remember that there are brain changes in adolescence, and you've heard some about this already. It used to be several decades ago, and even maybe more recently, we thought brain didn't change much in adolescence. I know when I did my training, you thought two years old was kind of the last development of your brain. Not true, but actually brain changes, some precede pubertal and hormone body changes, some may be the consequence of pubertal hormone changes, or they may be totally independent of those processes. Okay, this shows an imaging slide. This one is just structural imaging of the brain. What I'd like you to focus on, it shows the wide age range of kids and young adults here. On the vertical axis, it's showing percent of total adult relative to total adult brain volume. And I just want you to focus on the highlighted arrows there that show you different structures of the brain mature at different ages. Now notice it's age on the bottom. Puberty isn't there. And that really is something lacking in the literature, that we really don't know how puberty affects those things. People tend to look at age. So I think the jury is kind of still out where puberty affects specific regions. Now, what about the health paradox of adolescence? Well, adolescence really is one of the healthiest and most resilient periods of the lifespan. So, pretty lucky to be an adolescent from that perspective. They have enhanced strength, speed, reaction time, reasoning ability. They have increased resistance to cold, to heat, to hunger, dehydration, most types of injury. Yet, the overall morbidity and mortality rates increase by 200 to 300% from childhood to late adolescence. I mean, that is really kind of an unacceptable percent change. What are some of the sources of these, of the morbidity and mortality? Well, primarily, causes of death and disability are related to problems with control of behavior and emotion. You know, you see increasing rates of accidents, of suicide, homicide, depression, substance use. There are also high rates of risk-taking, sensation-seeking, and emotionally influenced behavior. So those are some of the things happening in adolescence. Now, why might puberty matter for taking some of these risk behaviors to heart? I particularly like this view that Ron Dahl, one of my colleagues when I was at Pittsburgh, has. He likens puberty or early puberty to starting the engines without a skilled driver. And I think that's pretty accurate. So earlier timing of puberty results in several years where an has this totally mature body and sex-activated brain circuits, yet they're really fairly immature in terms of neurobehavioral systems that are really necessary to control those behaviors and for affect regulation. Remember, those are the things that we just talked about are key with these risk-taking behaviors. So what's likely to happen? Well, perhaps increased risk for disorders of self-control, difficulties navigating complex social, emotional situations. So how many people are stressed out now thinking about their own adolescence or the ones they have at home, thinking, oh my gosh. Oh, few hands, so. Okay, let's think a little bit about the impact that stress may have in adolescence. And children with adverse early life histories have double the risk of emotional disorders from childhood to adolescence. So we've heard some about pregnancy risks, some about prenatal, neonatal risks. The second thing here is that there is calibration of stress-mediating systems, these stress axes you've heard about today, that occur earlier in life. Increase in activity of the stress system with puberty may contribute to this vulnerability that I talked about earlier and to negative outcomes in adolescence. Okay, in rodent models, and much of what we know for this is related to different lower animal models, we know that the period surrounding puberty may be another period when HPA axis is programmed. We heard from Dr. Vadva in terms of programming of HPA axis. That was in the neonatal period. So we're now thinking during the biological transition of puberty and adolescence. So improved conditions in puberty can reduce the impact of early life stress on emotion and stress reactive systems. So stressors encountered in puberty have strong long-term and negative impacts. Again, we know more in the animal model, but certainly your studies are going to shed a lot more light on that. This is some work done again in the rodent. On the left-hand side, for the vertical axis, it's looking at corticosterone. In humans, it would be cortisol. A restraint stress with the black horizontal bar. And you can see on the left block, prepubertal and adult rodents have virtually identical stress responses to a restraint stress. But if you look at a chronic stress situation on the right block, you can see tremendous differences with the prepubertal not mounting as high a stress response. So there are developmental changes in stress-sensitive brain regions in puberty. that may make an individual more vulnerable to some harm, but here I think is a great key to think of. It also can allow for opportunities or interventions to diminish earlier emotional or physical trauma. So the opportunity for intervention. Now, Dr. Chrousos laid out a great list of how stress can impact physical health. Today, I'm focusing a little bit more on mental health and psychosocial problems, but some examples of physical health issues. Dr. Chrousos, actually, I thought you were going to show a modification of this slide, but this comes from Chrousos and Gold work showing different HPA axis responses. And what I have highlighted in red are the ones that you see either initiated or peak during adolescence. Now, certainly there are more we could list here, but for example, depression and anorexia nervosa, we know those occur more during the adolescent period. So the red means adolescent in this case. Now, what's the impact of context on the stress system? I'd like to give you a few examples from the child and adult research child and adolescent research that is out there. And first, I'd like you to keep in mind what Selye has said, which is every stress leaves an indelible scar, and the organism pays for its survival after a stressful situation by becoming a little older. So hopefully there's a good side to this too. Now, what I'd like to show you today is thinking about different contexts that we've looked at the HPA axis response in, or even basal conditions, using some child and adolescent examples. For example, children from orphanages, child maltreatment. We heard that that's one of the highest ranked stressors that I think we all would agree with. Kids with obesity, also foster children. I'm not going to be showing anything about PTSD or depression because I'm thinking that the people tomorrow will be doing some of that. But those could also fit here as examples. So thinking about kids in orphanages, this was an earlier study from Megan Gunner. On the bottom, first of all, on the vertical axis, it's looking at daily average cortisol done across 3 days in children. Romanian orphanage kids are on the left, early adopted children in the center, and then Canadian-born children that were just like your healthy comparison group of kids. You can see for their average cortisol, there are big differences here. So the Romanian-born children who then were adopted but later have much higher cortisol levels. What about in the daycare setting? This one I'm using mostly to illustrate gender differences. Dr. Chrousos mentioned gender at the end of his talk here, and there truly are a lot of gender differences in stress research. What we're looking at here are boys and girls, boys in the solid line, girls in the dashed, and the top figure is focusing on anxious and vigilant behavior. So you can see that girls on this, in the lower 25th percentile at the left of anxious or vigilant behavior, and boys have essentially the same cortisol concentrations. This is during a stress paradigm. You get huge differences when you're at the high part, 75th percentile or greater, of anxious or vigilant behavior. Broad gender differences here. And it essentially flip-flops in the bottom slide. The bottom slide is focusing on angry and aggressive behavior, where boys, you see the higher cortisol with the higher aggressive behavior. So big gender differences in this area. What about child maltreatment? For maltreatment, I'm talking about either physical, emotional, or sexual abuse. So in some studies, they combine those 3 things. Some talk just about sexually abused, so it does vary. This is a slide that Dr. DeBellis and I were fellows with Dr. Chrousos. It's on the left, we're looking at plasma ACTH during a physiological stressor. And you can see in the filled in circles, those are the abused girls. They have a much lower response to this physiological stressor than a healthy comparison group. And also, even before the stressor, the CRH test, there are differences here. So already at a young age of these girls were roughly 6 to 11 or 12, you see some big HPA axis differences. Now, this was when girls were younger, but the next slide I'm going to show you is from the same group of girls, but it's following them longitudinally. And this is really quite a unique study. The principal investigators were Frank Putnam and Penny Trickett and Jenny Knoll. In this slide, across the horizontal axis, it's showing years since CPS, Child Protective Services, substantiation of abuse. We're looking at non-stress cortisol concentrations, and you can see at the early years, which was at the beginning of the study, the sexually abused girls in the dashed line had a little bit higher cortisol concentrations. As you go out in time, you see differences in cortisol where they have more of a blunting the longer out the abuse had from the situation of abuse. So this is really a very unique study. Right now, these girls are now young women in their 40s and with a proposed follow-up of this. So I would say stay tuned for some more information on this. This is the same sample, but it's looking at obesity. Focus on the far right-hand part of this, where in red you see abused girls. About 40% of them meet obese criteria compared to about 30 in the healthy comparison. You don't see this significant difference until young adulthood. So some of the things that you were mentioning, Dr. Vadba, is they might not occur right at birth, but you're seeing longer term changes here. What about how might marital function or parenting impact child stress responses? On the left, we're looking at wake-up and bedtime cortisol. Bedtime cortisol is always lower. And the solid line, MF, stands for parent marital functioning. So the higher that score is, the better the parent marital functioning is. So you can see that there's a wider spread at bedtime for those with higher maternal marital functioning. They have lower cortisol, and you want your cortisol to be lower at night, so that's good. If you go to the right-hand figure, it's looking at adolescents compared to those kindergartners, and you do see some differences, but the lines really are not that different depending on if you have low or high marital functioning. Okay, how might early puberty or in early adolescent girls impact the stress reactivity and behavior problems? Focus just on the left-hand side of this slide, and what we have is rule-breaking behavior and cortisol reactivity. In the dark bar, you see boys with early timing of puberty who have low cortisol reactivity, the far left bar, have much higher rule-breaking activity. So here you have kind of a difference with early timing versus on time or late, and the cortisol reactivity variable. So timing does matter. In this study that was by Sussman and her colleagues, there were no effects for girls similar to this. So I want to focus now on more of a physical health outcome, where we look at the impact of the stress axis on bone. You've heard a little bit about osteoporosis being one of the later health issues that we're concerned with in terms of stress. This figure is taken from a study done at the NIH a number of years ago. It's looking at bone mineral density in women on the x-axis, and it's showing two groups, depressed women versus normal, healthy women. And in each of the 6 areas of the bone, you can see that the depressed women have lower bone density in most of those. Now, what we think this is related to is the HPA axis. In fact, these women, if I'm remembering correctly, had higher urine cortisol levels, so that would be an indicator of the stress response. Now, I was able to do a study in adolescent girls. In adolescence, girls gain accrue about 50% of their bone mineral density. So it's really a crucial time. So this is in 262 adolescents, where we looked at the impact of depression and anxiety symptoms on bone health, as well as smoking effects for that. And what we found is girls with higher depressive and anxiety symptoms, it was associated with lower bone mass in these girls at the baseline visit. And how that relationship varied depended on what kind of a smoker you were, never smoker or ever smoker. So we were encouraged from a scientific perspective that we saw excuse me, something similar to the adult literature, but it's really worrisome from a health perspective to think that depressive symptoms or substance use might be impacting girls at this really kind of risky period. So we found that 1 to 6.5% difference in bone mass, depending on where you were on the depression or anxiety scale. And those percentages are equivalent to what a postmenopausal woman would lose annually, 2 to 5%, or a woman on Depo-Provera, which is a hormone contraceptive. So girls potentially, adolescent girls, are having a deficit in their bone bank. I think I'm getting my water rather than yours. Now, we haven't mentioned intervention and what that might look like with changing the stress axis. This is in foster children who are 3 to 6 years of age. And in the top left, the RFC stands for regular foster care. The panel to the right is the same type of children in foster care, but with this really fancy embellished program, both for the parents, the kids, and their whole environment. And the bottom left is just a healthy comparison group of matched kids. Now, this is looking at AM/PM cortisol differences, so it focuses more on diurnal type concerns. What you want to have is you'd like to look like this bottom left figure. And the kids in the enriched environments, cortisol across time, across a 12-month period, the enriched environment is very similar to healthy controls, which is good news. But the regular foster care kids do decline in what their diurnal rhythm is. Now, or difference, I should say. Now, these are kids who have probably been in an average of 5 foster care homes, so certainly stressful for that. So intervention seems to change the HPA axis for these kids. I'm putting up the same biological transition slide to remind you about the vulnerability for some individuals, but that the context may moderate how you do. But there is a silver lining. Even though you can be especially vulnerable to harm at this time, it also allows for the opportunities or interventions that I mentioned, and I'd like to elaborate on that a little bit. For example, we heard stress is a part of everyday life, but as parents or educators or clinicians, We need to think about things like how does the negative context impact a kid and can it be changed? And then recognizing emotional stress in these kids. Not everybody can recognize that. Can we intervene when necessary and also promote successful coping strategies? Some of these do have some policy implications, but let me elaborate on this a little bit. In the family setting, I think we could think about things like safe home environment. Is that around? How can you minimize parent discord or enhance parent mental health or their knowledge and communication about child development and communication with their children? For schools, thinking about the education for teachers about stress, education for safety issues about bullying. Not a day goes by that we don't hear about the impact of bullying on children and adolescents. Also in schools, are there opportunities for activities and relaxation? You mentioned some of the studies with relaxation in school systems, which seems like a good idea. In neighborhoods, are there safety issues in certain neighborhoods? Yes, we know that. Is there opportunity for recreation? All of these things could potentially decrease stress. And then I'd also like to think about what I call the setting of technology. All of the different technology kids use, from texting to Facebook, all of those different things. Do adults have the understanding of this technology, and can they monitor what's going on? Because certainly those sorts of things can be a stressor depending what kids are texting to each other. So I don't think our work is done. We need, in this area, we need many more longitudinal studies that tell us more about the impact of these early stressors or current stressors on children and adolescents. We're also really lacking diverse samples by race, by SES. We know virtually nothing about kids in third world countries and cortisol and stress. We can only kind of surmise from those sorts of things. Collaborative studies where disciplines are included, behavior studies including biology, biology including behavior, and then culture and context are included in that as well. That's it. Perfect timing. Sure, thank you.
We have time for a couple of questions before our break.
Oh, there's one over there.
Dr. Brown.
As I'm listening to all these factors that impact stress, let's just take adolescence. Is there any evidence that increased understanding on the part of the adolescent as to these various factors would facilitate their capacity to deal with all of them? And I'm not even sure which of the many things you've talked about, but if there were one or two, like, I don't know, expression of aggressiveness or something, if you knew it was in your brain or your, you know, what's that relationship?
I mean, I do think that's kind of the $64,000 question. You know, like from a parent's view or a clinician's view, you might say, I told her that that was really chancy to do. But, you know, if your friends are all doing that and, you know, they're encouraging you and that sort of thing, I don't think education only is the answer. You know, I think there's so many factors that go into it. Because and also because of the brain immaturity too, and the mismatch between all those things. But
yeah.
Does that kind of answer, or?
I wasn't thinking parental admonition, because we know there are all sorts of problems with that, but just as you, or I did anyway, study science in 7th grade or something. We studied machines, I remember that, but suppose you had studied something about impulse control and the elements that affect that in your own physiology? Is there any studies that would suggest that kind of knowledge would have an impact?
You know, I haven't seen any. I don't know if anyone in the audience has, but I think we could do a better job of exciting kids about, for example, about the brain and brain development. You know, I don't know how many health classes show some of the colorful pictures that you showed. You know, that's more exciting to a kid, or thinking about imaging studies and that kind of thing. Like, I do know there are classes in school about like relational aggression, which has some to do with bullying. So how much of it sinks in, I don't know, and I'm not aware that anybody has studied if that helps.
Is it true that the majority of the adolescents are doing well adapted?
I'm glad you brought that up, George, because that is very important. There are some studies that show that it's really only 20% or less of all the adolescents that really have traumatic times during adolescence. So it's not all bad. There's a lot of good adolescents out there, and some of these risky type behaviors are very short-lived. You know, it's trying it, it's experimenting. So it's a small portion that go on to being the more problematic societal concerns. So, good.
Great.
Good. Thank you very much.
Thank you. 15-minute break. It's time to continue with our next speaker, Dr. Philip Gold from the National Institute of Mental Health, longtime collaborator, and a super expert on depression, stress and depression.
I was told to keep the talk really quite accessible, but I thought to myself, you know, this stuff is very easy, because when I was about 15 years ago, there were, you know, the New Yorker has little blurbs of newspaper clippings, and they had this one. Mr. Philip Gold was awarded the Kurt Richter Prize for the best neuroendocrinology by an investigator under 10. So I went downhill from there, I think. So I'm going to talk about depression as a dysregulation of the stress response. And I'm going to cover part of the stress response that really seems to be most involved with depression, although George very eloquently went beyond this into multiple systems. The World Health Organization has named major depression the fourth leading cause of disability worldwide. And I think this is for many reasons. We now know that depression is associated with increased systemic illness and a shortened life, but also depression is a serious illness that has a unique incidence in children and adolescents, and it's a lifetime disorder. So there are many people out there with this chronic illness that begins even in childhood or adolescence. The mental anguish of depression is only the tip of the iceberg of a serious systemic illness that doubles mortality at any age, independent of suicide, smoking, and other risk factors for poor health, including hypertension as well. The excess mortality is equivalent to approximately 9 or 10 years and is similar to that in stopping smoking and treating hypertension effectively. And patients with major depression have premature coronary artery disease and premature osteoporosis. And for coronary artery disease, the incidence is doubled, and osteoporosis is found, we've reported that even premenopausal women with major depression develop premature osteoporosis. Now, I thought I'd start this, you know, we talk about sort of traditional stressors with just sort of the kinds of existential things that make life difficult, even if you live a very successful and comfortable life. You know, we all face internal contradictions, ambiguities, and paradoxes. There are elements of victory and defeat and victory. There's loss of opportunity embedded in every choice. You choose one and you give up all the others. There's inevitable clashes between passion and duty. The liability to change in fortune, no matter how worthy. We sometimes must act in ignorance. That renunciation is intermingled with gratification and honor. There's the possibility of suffering while learning or changing, and sometimes suffering is necessary to learn or change. Coexistence of love and anger, the unreachability of the ideal self, the debilitating effects of perfectionism. Time is irrevocable. We cannot recapture the path, and life is a continuous progression towards death. So there are many sort of, to be alive, I think, is to have to deal internally with many conflicts that few, if or any of us, can escape. The stress system, one of the principal elements of the stress system is anxiety. Anxiety is essential for survival. If a person were being pursued by a bear, certainly would require anxiety to motivate him to find safety. Now, emotional memories, like those that are associated with anxiety, are exceptionally well remembered. They don't just occur for a moment, but they get encoded in a number of brain regions, but especially the amygdala. And they are very difficult to extinguish. Now, there are actually very good people who are working with medications that promote extinction of aversive behavior, of aversive memory, that would be good for post-traumatic stress disorder. Emotional memory can be retained even if other aspects of memory remain unchecked. And for instance, there's a story of a woman in a hospital who had brain damage to the part of the brain that affected procedural memory for objects and places and events. And he came by and he gave her a little pinprick every morning in her palm. And he came by 6 or 7 times, and each time she would withdraw her hand. And then on the 8th day, he pricked her, and she jumped back. And he said, by the way, do you know who I am? She said, I've never seen you before. So emotional memory can be retained even if other forms of memory are not intact, remain intact, rather. Evolution has given us an abundant circuitry to encode anxiety. And there are many, many more neurons that encode anxiety and many more fibers that are sent from the areas of the brain that modulate the thinking part of the brain. So there's just not much representation. And in contrast, there are not many fibers that go down from the cortex to the areas of the brain that generate anxiety. And it's very difficult to treat. And I'm really pleased to hear these methodologies that folks are developing to deal with anxiety and similar problems. During a stress response, so you have anxiety. The next is an inhibition of so-called vegetative functions whose activation would impede adaptive responses during threatening situations. That would be sleep, for instance. You don't want to fall asleep if you're being chased by a bear. Feeding, you don't want to stop and eat. Sexual activity, you wouldn't want to stop for that. And actually the capacity to experience pleasure is also somewhat clamped because you wouldn't want an individual who is running for his life to stop and pause and see something that gave some pleasure that was very interesting to him. And the programs of growth and reproduction are also sacrificed to a certain extent to provide energy for other sites. Now, stress hormone secretion, actually I'm just talking now about in general. There are 2 major stress hormones. You've heard a lot about cortisol, and also, rather 3, adrenaline and noradrenaline is also important. And they're both secreted during stressful situations. Noradrenaline first, and then cortisol follows. And but cortisol has many positive effects in normal concentrations, but it is very deleterious when the levels are high. It promotes the breakdown of tissue, muscle. It increases hypertension. It is an immunosuppressant. And there are many things that would be deleterious if one had sustained hypercortisolism. Norepinephrine is the same. It's treated from it's released from the sympathetic nervous system, and it promotes vasoconstriction, so blood pressure rate rises. It promotes increase in heart rate, increase in blood pressure, and increase in the contractility of the heart. But again, going on for a relatively longer period of time than one that is usually experienced during an ordinary stress response would cause many difficulties. Now, these stress hormones do things other than affect physiology, and of course they have effects on behavior as well, but they increase the intensity and the duration of emotionally charged memories. So that's a problem if you have a very significant emotionally charged memory and you're likely to be secreting cortisol and norepinephrine. Clear studies show that if you can prevent their rise to very substantial levels, then the aversively charged memory is more easy to extinguish. And it seems to inhibit access to positively charged memory. Later on in depression, it seems that, I thought this years ago, that depression seemed to be a continuous access to negatively charged memories that were without the content, which were influencing or producing the psychological pain. There's adaptive redirection of energy. I mentioned that. Breakdown in proteins, so muscle is broken down, and the amino acids are incorporated into new proteins that can be served as fuel. And there's mobilization of glucose for the brain. So your blood sugar goes up when you're stressed. I might stop for a moment to talk a little bit about the mechanism of that, because it's interesting, because everybody hears so much about insulin resistance, and it's very simple, really, that when you're stressed, cortisol causes a block in the doorway from the blood into the cell for insulin. So ordinarily, insulin will excite the receptor and enter the cell and exert regulatory properties and lower blood sugar. And what happens during stress is the cortisol is secreted, so more insulin has to knock on that door. Higher levels have to knock on that door to get through it, because the door is now more resistant to being opened. And insulin has many deleterious properties. I'll talk about that later, but it increases the sympathetic nervous system. It promotes inflammation, promotes a bad lipid profile, and so forth. Now, there are other events that most of us don't associate with a stress response. And by the way, by stress response, I talk about running from a bear, but the same apparatus is called into play if someone hurts our feelings, or if there's a loss and we haven't adapted to it. So to a psychological stressor or to a physical stressor, inflammation is produced, and we become slightly in a pro-inflammatory state. And kids, for instance, who are not massively obese but who are heavy, they've done a study in 12-year-olds, they are all in a pro-inflammatory state. They all have increased levels of hormones that work on the immune system to jog its function, and these are called cytokines. One other new development is that new neurons are produced. The brain was not thought to divide. It was thought to retain a stable number of neurons, and they didn't change, so that one would not lose sight of one's personal history and memories throughout a lifetime. But we now know that new neurons are created. There is a so-called neurogenesis. And they are helpful in modulating the stress system. If you take an experimental animal, a rat, and stress the rat very strongly, the level of these compounds falls, and they're restored by the administration of antidepressants. And so one theory now is that antidepressants may work on in helping them to produce new neurons. And as I'll show later, depression is not only a disorder of physiology, but there is neurotoxicity, loss of tissue in many parts of the brain that are involved in a total symptom complex of major depression. But we never would have thought of that maybe 20 years ago. And nerve growth factors must be secreted also to promote the system to respond to stress. They're trophic to specific systems, and they coordinate repair. Also, rapid changes are required, and we call this neuroplasticity. If you stress an animal, if you imagine a neuron, you've seen them with the little strings that come out of it, or it will grow bushy outgrowths, and it will occupy more room, but its actual more, shape changes and its function changes. It is much more able to influence a lot more nerves and genes than the neuron was able to do before. Now, animal models of depression show that this neuroplasticity, it's like a tree kind of blooming and opening its limbs. So that if animal models of depression show that these nerve growth factors fall, and that if you give animals standard antidepressants, the nerve factors return, so that treating an established depression, even experimentally, it's not really depression in the rat, but it's a model, and it makes sense. It's a stress model that you can promote the regulation of the stress response. Because one of the things that happens pathologically is that it doesn't turn off. It's all right, it's good for us for the short term, but it gets to be a problem when it remains turned on. Same thing for autoimmune disorders. If we need immunity, but patients who have increased immunity are not healthier. These immunologic factors attack tissues and destroy them and cause diseases like rheumatoid arthritis. Now let me talk about major depression. I have here that it's both a disorder of mood as well as a serious CNS-driven systemic disorder associated with premature cardiovascular disease, pathologic bone growth, and death. I brought that up. I won't stop with CRH now because I don't think I'll have enough time. Excuse me. Now, what is the composite stress response that I've told you about? They're fear-related behaviors and anxiety. There's an inflexible mood and cognition. You don't want, if an organism is in danger or if we're in danger, we don't want to be having our ideas and thoughts jump around from one place to the other. Or even working on complex associative programs because there's not going to be time to execute them in the context of a life-threatening situation. So the mood gets clamped in this stressed mode. You see, they get stress hormone production, and they get blood, which is rather sugar or blood sugar, which is not going into the cell sufficiently because the door is fairly locked. Therefore, you get glucose for the brain for the nutrients that the brain needs in order to function properly. There's an inhibition of neurovegetative programs, as I mentioned, sleep, feeding, sexual behavior, pleasure. Now, the depression I'm presenting here is a dysregulation of that stress system. As I mentioned, the stress system is fine if it turns on momentarily or for a few hours or even a few days. But if it stays activated in one way or the other over months, then tissue damage and illness results. just as the illness responds to autoimmune disorder. Now here's a phenotype. A phenotype is a form of depression. We generally think there are many different forms of depression, but there are two that are recognized formally. And one, this one called melancholic depression, is really one that holds true. It's very similar from one patient to the other. Rather, in other situations, the depressions seem to be heterogeneous. So this form of depression belies the term depression because it's a state of activation, arousal, rumination. It's often associated with anxiety, and as I mentioned before, that anxiety is attached to the self. And the cruelest manifestation of this particular form of depression is that people lose their regard for themselves and regard for what they were in the past, in their past achievements, what they are in the present, and what their prospects are for future gratification for such an impaired self. There are physiologic indices of hyperarousal. The stress hormones are elevated, but the inflammatory mediators are elevated. Also, one other thing I didn't mention, there's also more blood clotting. The reason I think, by the way, going back to inflammation, that there is inflammation during stress is there's a premonitory rise in a fight or flight situation. In case there's an injury, there are inflammatory mediators around to repair the damage. And the reason that, pardon me, I'll go back on here. Okay. Now, the depressed mood and melancholy, I've described that to you in several different ways. It intrudes upon elements that define our humanity. The cardinal manifestation of melancholia is a pronounced sense of worthlessness. The anxiety is an important part of melancholia, and it is directed against the self. It really is an anxiety about the self. I'm worthless. No one will care about me. There's intense rumination. And this is an intriguing feature which is present in almost all patients with melancholia. The mood is more severe in the morning and the evening. And it may just, for the most part, get only slightly less severe, but that pattern holds true. And the mood is clamped like it is in the stress response, but in a pathologic way. There are cognitive deficits in patients with depression also. There is a sort of a switch from sequence, associational type learning and executions to more well-rehearsed, almost instinctive programs that the organism or the person can put into play rapidly in order to survive. But in a sense, these two forms of cognition hold true in the person who becomes sustained depression. And one of the issues, disturbing issues in patients with major depression is that memory and intellectual acuity is somewhat influenced by the depression, and it's completely reversible. And melancholic depression, also I had mentioned earlier, seems like an almost continuous bombardment of emotional memory that had been laid down. And this emotional memory, by the way, comes back without the content. You feel the feeling, but you're not able to put your finger on the fact that this is because someone had insulted me terribly 25 years ago. Now, there are neurovegetative features in depression, in melancholic depression. Instead of inhibition of sleep, there's a sleep disturbance. And that sleep is almost always an early morning awakening. If I see someone who's depressed and they wake up early in the morning, I think the diagnosis is fundamentally made. There's a loss of appetite. There's reduced sexual activity. There are decreased endocrine programs for growth and reproduction. So it's similar to this vegetative picture in stress, but it's dysregulated. There's a maladaptive direction of energy. I mentioned there's an excessive breakdown of protein, and there are high blood sugar levels because of insulin resistance, which occurs early on in the depression. There's excessive cortisol secretion and excessive norepinephrine secretion. Here's the data taken in our laboratory a long time ago, and if you look over in sort of the left panel, which is at night, that's what those dark columns represent, the levels in red in the depressed patients are significantly higher than they are in the controls. And that is through the night, which is intriguing because it suggests that these high cortisol levels are not necessarily related to the stress of the illness during the daytime. And norepinephrine secretion is also increased. This is an interesting kind of study. We had patients and we measured their baseline norepinephrine. This was spillover in the arteries. And we followed them early on, and the blip you see is when the control subject was playing a simple video game. But norepinephrine went way up, and it remained high. The lower panel, the black curve, is the baseline level. And it's interesting that even though there's an increased baseline level, you can still jack the system up. Usually, if you have a system that's activated, it's a little harder to further jack it up because the mediators that influence it or raise it become less sensitive. And I mentioned the effect of stress hormones encoding emotional memories. And in depression, it's excessive inflammation, and it's an excessive, actually I didn't mention this, it's excessive increase in blood clotting also. Just as the inflammation is premonitory to perhaps protecting the person if there's a possible injury, the increased coagulation is there as a help or a stay against potential hemorrhage. And now here is a study we did where we put in a little cannula and took bloods out at regular intervals for something called IL-6. IL-6 is a cytokine. It's really one of the most important mediators that stimulates the immune system. And it also stimulates what is something called the acute phase response. We see that it is elevated throughout the night and becomes rather during the daytime. It falls at night. There are certain mathematical formula you can play with these kind of curves and see if they show a regularity or if they show a sort of an irregularity that is dictated, though, by a physiologic system. So the levels were not only high, but this form of secretion, the pattern of it, was also abnormal. Fibrinogen is one of the principal clotting factors. And just if you measure a depressed patient at baseline, you'll see that the fibrinogen levels are elevated. And again, if this were stress, and they're elevated during stress as well, this would be premonitory, perhaps as a stay against hemorrhage. Now, a little bit about the circuitry. Much more is known about the circuitry now. There are 7 or 8 areas of the brain where there is a notable loss of tissue that impairs the functions of elements that are involved in depression. Now, at the top, the green arrow is pointing to the medial prefrontal cortex. And the medial prefrontal cortex is a very important function. It restrains the amygdala fear system. It promotes intellectual-like thinking. It restrains the stress hormones. And perhaps its most important function is, as I mentioned, is restraining the amygdala. So when this break on the amygdala, it has diminished function. Amygdala function increases, and there's increased anxiety. And I think when that happens over the long haul, that's what produces a depression. The amygdala, by the way, I'll show you next, is the area of the brain that is essential both for the conscious experience of fear, but also it prints memories that are not necessarily connected with specific sources and stores them for long periods of time. So there's the amygdala. It is essential for fear, conditioned fear responses, inhibition of the medial prefrontal cortex, and activation of the CRH and locus coeruleus systems. So by the way, there's a positive feedback response going on. You want the amygdala to be calm so there isn't anxiety. And I think sometimes depression is an organized state of anxiety, but it's attached to the self. So you want to restrain the amygdala. Now the medial prefrontal cortex sustains it, but when it's low, the amygdala function increases. And then the amygdala function then goes on to stimulate the medial prefrontal cortex more. And it goes on to, it sets into motion a positive feedback loop. And these systems are the systems that control norepinephrine and CRH secretion. And they also influence the medial prefrontal cortex in a positive feedback loop. The amygdala with a positive feedback loop. So this system is really whirring around once it gets started. It's very hard to stop it because each element is not only playing its own role, but it's influencing the functional activity of the others. Here is a graph of the tissue volume which is taken from a medial prefrontal cortex. And rather, this was taken during a PET scan. And what we see is the volume of the prefrontal cortex is rather high in the control. It's reduced in both the bipolar and the unipolar patients. And in bipolar and unipolar patients, the decrease in the volume of it's called the subgenual prefrontal cortex, is as much as 40%, which is enormous. Now, there's some suggestion that antidepressants can reverse this, or mood stabilizers can reverse this, and the data will be coming further. I think it looks like they do reverse the loss, so it's not irrevocable. It's another important reason to treat depression, I think, with medication, because the medication can restore some of these pernicious processes back to their homeostatic state, as George described it. And there have been some imaging studies in depression that sort of interesting, and I'll just show you three that suggest that the medial prefrontal cortex activity is not functioning as well, and that the amygdala is overactive. Here, one study which was very interesting, faces with distinct features were flashed in a very rapid function, and went from a very smiling expression down to a neutral expression and down to a frowning expression. Now, the pictures were being presented too quickly to be seen by the naked eye. But these folks were hooked up to the imaging machine, and what you saw is that the depressed patients started to increase the activity of their amygdala just before it got down to neutral. It didn't have to go down into the frown, and the controls had to go rather far down to see the frown, so that the pregeneral cortex function was not proper. By the way, one of the important functions of the pregeneral prefrontal cortex is estimating the likelihood of punishment or reward in a particular situation, which I think you would see as very important in a depressive disorder. Now, when patients with major depression, anticipate a negative event, a circumstance that is often described as dread, their subgenital prefrontal cortex dysfunction will lead to an abnormal, persistent, negative affective response, even if the dreaded response doesn't occur. So the depressed patient can be told something terrible is going to happen, and when it doesn't happen in 2 or 3 hours, the controls are back to normal. The depressed patients are still functioning as if they are expecting a punishment rather than reward or a neutral situation. When depressed patients reflect on their internal state or on words that apply to themselves, the subgenual prefrontal cortex activity changes, indicating significantly more negativity than controls. And patients with major depression have a significantly lower response in the nucleus accumbens to a pleasurable stimulus. The nucleus accumbens is a dopamine-rich area that plays a great role in transducing pleasurable experiences. So if you give a pleasurable picture, say, or a series of pleasurable pictures to controls, their nucleus accumbens will significantly increase in metabolic activity. And in the depressed patients, it's much less evident. So there's this anhedonia, or difficulty in experienced pleasure. There's another form of depression. I'm not going to spend a great deal of time in this. But remember, you have melancholic depression with its kind of its arousal. The atypical depression is associated with a pathological hypoactivity. It's associated with profound lethargy and fatigue. Patients feel much less alive than usual rather than anxious. They have, like the melancholics, a decreased capacity to experience pleasure. They have an increased appetite. They have increased sleep. They have a deep sense of loneliness as a part of this disorder. I think because one of the things that happens is that whereas maybe the depressed patients have too much of capacity to to retrieve negatively charged emotions, these patients seem to be out of touch with themselves, and they can't seem to retrieve memories, emotional memories, and they feel empty and alone. Because all the figures in their life, sort of figuratively, whom they should remember, are not accessible to them. It's as if they lost their past. The severity of depression here is the antithesis of that in melancholia. It is less in the morning and more severe in the evening. And as I mentioned, often there's a state of loneliness and isolation. We've done studies in these patients. I won't show you the data. But these patients actually have evidence of a hypoactive HPA axis. Their baseline cortisol are low. We do certain stimulation tests that give us information about the relative roles of the brain, the pituitary, and the adrenal glands in regulating cortisol secretion. And it seems that at the level of the brain, the major stimulus, which is an activating compound, is significantly lower in these patients than in controls. We have other sets of data, by the way, where the response to other stimuli are also hypoactive. You get a big cortisol and ACTH rise with graded treadmill exercise, and this doesn't occur as readily in the patients. And the pattern, by the way, was compatible with a central rather than a peripheral defect. It's a little harder to treat than melancholia. Melancholia has probably the highest rate of response among the depressions, and it responds to a range of depressions. This drug is not used very often, although it still has its place. But imipramine, which is a tricyclic antidepressant, is great for melancholic depression. But atypicals don't respond to it at all. They respond to it less than they do to placebo, so it probably has an aversive effect on them. Perhaps it turns down the axis even further. I mentioned this before, that the melancholic patient seems to be in a continuous contact with the past, the aversive past of the memory, and the atypical depression seems to have lost his past self. Now, I mentioned the medical complications of major depression, that the major depressed patients have a increased risk of mortality, twice that of healthy controls, increased premature coronary artery disease, and osteoporosis. So here, osteoporosis, I don't think you can see this slide very well. On the bottom, if you see that dotted line, it intersects that normal range right at the level that would denote osteoporosis. But I didn't get a good figure for that. We've actually done some bone biopsies in women with osteoporosis, and here's one where you have the normal bone on the left and the abnormal bone on the right. And this is in a 38-year-old woman. And what you see over there, you see these canals as they were, and a continuous connection between these structural components. So it adds stability to them. But here, that structural component is lost. Young women don't tend to get fractures in their 30s or 40s because they're young and their bone strength is still strong. But when they're 50 or 55, rather than 65 or 70, they're very prone to osteoporosis. It's important to recognize that, and I think many doctors will think about bone health in patients with major depression because it can be so readily treated. Coronary artery disease, I'll flip these by very quickly. These are studies on norepinephrine secretion and epinephrine secretion in patients with major depression. These were very severely depressed patients. As you see, it's elevated. This is in the cerebral spinal fluid. We actually had a lumbar drain in several patients that we can excuse CSF, cerebral spinal fluid, for 24 hours. It's a virtually painless procedure and perfectly safe. So you see in red, the depressed patients are significantly elevated above the controls in black. The next pane on the top is plasma norepinephrine. And one thing I would note here that the levels still go down at night, but they reach their peak at around 9 or 10 o'clock, both in controls and in patients. And it's a very cardiotoxic compound, and that may be one reason that the time of the greatest risk for an acute heart attack or sudden death is around 9 or 10 o'clock. And the next one shows again elevation of the norepinephrine in the patients versus the controls. And the last is epinephrine. And epinephrine doesn't show the same diurnal variation, but it's much higher than in depression. And it's a cardiotoxic compound too. And we also show that cortisol follows these same patterns. And cortisol and adrenaline and noradrenaline would act quite synergistically in being cardiotoxic. And they are simultaneously elevated at the same time. And it so turns out that the highest levels of adrenaline are at that time as well. These patients were very severely depressed. There's some suggestion that depressed patients do have high blood pressure, but their control blood pressure is in yellow, and in red, it's again that's in the patients. And then after these patients were so severely depressed that they got ECT, but after ECT, the systolic tension fell back again to normal levels. And here we have the diastolic pressure, that's the lower number. It's also up prior to the ECT, but comes down. Now, they're elevating fasting insulin and glucose levels in patients with major depression. And we matched these people very tightly, BMI matched controls. And we had 32 patients. They were thin, so that obesity was not a compounding factor. And what you see is that they have increased levels of glucose and increased levels of insulin, increased levels of glucose. These levels do not meet criteria for insulin resistance or type 2 diabetes, you need to get the fasting blood sugar. Now it's up to 100 rather than 120. But they are significantly higher, and there are data that there is sort of a continuous risk with diabetes with respect to the levels. So if the levels rise even 5 points over 10 years, and that's carefully documented, there's a doubling in the incidence of type 2 diabetes. And patients do have type 2 diabetes 2 or 3 times more frequently than controls. And again, the high insulin levels are pernicious, with insulin causing increased sympathetic activation, raising cortisol, causes increased, pardon me, I'll skip that. Couple more. Let me get this to go back. Okay, oh, here's an interesting slide. These two women are the exact same weight. They have the same BMI. Let's say it's probably around 125 or 30 pounds. But this patient, who's a depressed patient, has a much larger mass of what is called abdominal fat, intra-abdominal fat, which is different than the subcutaneous fat. And as you see, this intra-abdominal fat is producing all kinds of inflammatory mediators. All of these are compounds that generally activate their cytokine receptor. Now, with so much activity and increased inflammatory activity, these inflammatory mediators also raise insulin secretion, and insulin produces insulin stimulates the secretion of cytokines. So we have another positive feedback between metabolism and insulin. The reason this may be so, this is just a guess, it's because it was not really very clear why inflammation should be so wrapped up with blood sugar. But the thought is that with the increased what the increased levels of the cytokines do is to affect norepinephrine levels that will further increase the insulin resistance which you need. So there's a very tight relationship. Another thing that is possible is that when one gains weight and you have more sympathetic nervous system activity, that also can be soluble. Now, here are some composite stress responses of the controls, the patients with depression, and then the epinephrine findings. So the controls, for the composite stress response, you have these elements that are all changed in depression. Fear-related behaviors and anxiety, inflexible mood and cognition, stress hormone production, redirection of fuel to the bloodstream, premonitory inflammation and coagulation, inhibition of neurovegetative functions, which were the ones we talked about, sleep, and so forth. And also, there is nerve cell growth factor, and increased neuroplasticity, and there is effective termination of the response. And I didn't mention here that neurogenesis, or the production of neurons, new neurons, also occurs as part of the stress response. And in stressed individuals, or at least in stressed animals, as I mentioned, that the increased generation of neurons that submit growth factors, that means that the volume of the brain, and especially the subgenital prefrontal cortex, will be protected. Sorry about this. I was up most of the night, which I shouldn't have been. Now, melancholic depression looks very, very similar. You have increased and prolonged fear-related behaviors and anxiety, inflexible mood and cognition, increased and prolonged stress hormone production, increased redirection of fuel to the bloodstream and brain, increased and prolonged premonitory inflammation and coagulation, increased and prolonged inhibition of neurodegenerative function, and decreased new nerve growth cell factor with decreased neuroplasticity and insufficient termination of the response. The last is atypical depression, and again, it's the antithesis of major depression. As you see, and I've told you before, it's characterized by profound lethargy and fatigue. Patients feel less alive, a decreased capacity to experience pressure like the melancholics, increased appetite and sleep, a deep sense of loneliness. The severity of the depression is less in the morning than in the evening, and often a state of isolation and loneliness. Okay, so I'll stop here. Any questions? Or is that not?
Down there.
Yes, please.
And I'm wondering if that, what the relationship is the stress triggering an already existing bipolar syndrome?
Stress does precipitate bipolar disorder, but we understand less about the actual biology of the physiology and the biology of bipolar disorder. And it's one of the reasons you didn't see many slides on bipolar disorder. The hormones and the CSF compounds have not been examined as readily. It might be in part because of a relative dearth of bipolar patients, but also it's very difficult to study them when they're manic. I personally feel that they're part of the same illnesses, that there's a continuum on from depression, and it gets more severe, and perhaps there can be a development of bipolar disorder. And it seems that many patients who do have bipolar disorder had unipolar depression before they developed the bipolar depression. The good news about bipolar depression is that there are better treatments now with the anticonvulsants like lamotrigine and so forth.
Dr. Rosenthal.
Thank you very much for that comprehensive description and discussion. One thing that really intrigued me as we've both been dealing with depression for many decades now, which is completely new and unexpected, is the extreme loneliness that you describe in the atypical depressed people. Are they any lonelier than any other depressed people? I mean, where is the data?
No, I have no data. It's my clinical observation that I've seen that kind of malignant loneliness more frequently in the patients.
Could you sort of describe how how they would express it, or how it would emerge in a clinical interview?
First of all, they simply say they have. It's often very difficult for them to interact with other people, and some say, and they seem burdened by even the slightest task. And many tell me that they feel so depressed in a way that even carrying on a conversation seems like tremendous work. I don't know if you're
very familiar to me because, of course, one of the areas of that I've researched is seasonal affective disorder, and most of those people are atypically depressed.
Right.
And I think it's the effort of interpersonal engagement that is overwhelming, and that they can't bear it. And I've seen many people just let go of a relationship because it's just too much work.
You can't do it, yeah.
So I've never sort of seen that as loneliness, but rather as just, this is more work than I can muster and more work than it's worth. And if the partner or lover or friend has to go, then that's just what's going to have to happen because it's all I can manage.
No, I think that's right.
Yeah.
But I do think, this is just metaphorical. They do seem less alive, flatter. And I don't hear much talk from them about their past, as I do say from the melancholic patients who are equally depressed. They may be disconnected from it.
No, It's interesting, and I think that the melancholic depressives are more agitated and more
Yes,
activated.
they're activated.
And that goes along with the HPA axis
Right.
activation, and these people are just flat. I agree
Yeah,
with you. Thank you.
it's an activated or
Thank you.
continuous anxiety. Any other questions?
Dr. Romero?
Yeah. Phil,
Phil?
can I ask you, thank you very much for the presentation. I have 3 questions, very brief. One, you show a study in which interleukin-6 was elevated in the morning, and I saw that you had samples taken throughout the day. Does that represent a change in the circadian rhythm of interleukin-6?
Yes.
And how often did you sample the patients? Because
I think we were every 15 minutes on the
15
patient,
minutes.
Yeah.
The second question is, you know, forgive me for asking this, it's an obstetrical question. Most women have a change in mood, and then we have the extreme postpartum depression that can end in suicide. And I wonder, one, are there studies about what imaging, neuroimaging in postpartum depression?
I don't believe so. George and I have studied that, endocrinologically, and it seems like the very high cortisol levels they have because of the 3rd trimester plasma CRH levels. When that's removed, then they have an activation of the immune system.
It's an atypical form. In other words, cortisol goes down for 3 or 4 months after delivery.
Yes, hi.
David?
Hi, Phil, thanks. You've really pioneered this area. I appreciate your work. 2 questions. So is the elevated IL-6 in the morning related to the elevated CORT at night? Does CORT drive that, or is it independent of that?
CORT suppresses IL-6 actually for the most part, but IL-6 does stimulate CORT to a great extent. And that stimulation is probably for a negative feedback response. You have IL-6, which is causing inflammation. Yeah, and you have, it promotes the secretion of cortisol, and cortisol restrains the axis. So that's one component of cortisol's negative effect on the immune system.
Coincides with the catecholamines. So when the catecholamines are high, IL-6 is...
IL-6 is high.
But it's not
the evening is low.
It's not immediately obvious why it would happen, given the elevation of CORT, particularly at night in depression.
Because IL-6 and CORT are probably they don't have similar, ordinarily have similar circadian rhythms.
All right, thank you. So the second question has to do with medial prefrontal cortex, which you mentioned inhibits amygdala. Medial prefrontal cortex is also involved in social perception and cognition. And so I wonder if you have any comments about that, the isolation of depression and the failure to
That's a
inhibit.
good question. I don't think people have studied atypicals, but it wouldn't surprise me that they feel awkward, they feel like they're not going to present themselves effectively. That's a good question. I don't know if there's a modulation of a central sort of socializing component, which does exist.
Thank you.
It's could you speak to the long-term effects of living in a constant state of vigilance?
Good question. It hasn't been studied really as much as it should. And I think that there'll be the vulnerable population, like everything else, will respond with a depression, just like they'll respond with depression to various other things. But they all won't respond to the same thing. But that's not an answer to your question. I think people haven't really looked, or done a study to my knowledge, and it certainly should have been done, on what is the incidence of depression in people who've been chronically stressed. I'll bet I'm sure it's going to be higher.
No other questions? So thank you very much. And time to go to our last but not least speaker. Thank you, Phil.
Thanks, Phil.
Next speaker is Dr. Daniel Clough. He's going to talk about pain and fatigue states, PTSD, and war syndromes. Dr. Clough is professor of medicine at the University of Michigan in Ann Arbor, Michigan. and formerly of Georgetown University, and a collaborator and friend.
Thanks, George. The University of Michigan makes us show these slides. I'm not trying to show off drug company consulting. So you've heard today about the notion that there may be, if you will, vulnerable hosts that for one reason or another have some type of vulnerability that when they're exposed to stress. Phil and others talked a lot about the development of mood disorders. I was asked to talk about post-traumatic stress disorder, but I don't consider myself to be an expert on post-traumatic stress disorder, so I'm just going to talk very briefly about that. What I do know a lot about is pain, and so what I'm really going to talk about now is the role that stress has in triggering, exacerbating a number of different chronic pain states. So let me begin by being a little bit provocative. I'm a rheumatologist by training. I'm not a psychiatrist. I've become a neurobiologist as a result of studying pain. But these are knee x-rays from two different people. For those of you who might not be used to looking at knee x-rays, the bone at the top is the femur, the big bone at the bottom is the tibia. You can see an outline of the kneecap or the patella. And you see there the space between, on the left, between the femur and the tibia is actually made up of cartilage. Cartilage does not show up on an x-ray. Bone does show up on an x-ray. So as a rheumatologist or an orthopedist, when we look at a bone x-ray, especially a weight-bearing x-ray, we're looking for a joint space. And when we see a joint space, that indicates there's cartilage on the end of both bones, and that theoretically would be normal. But the question I have for you here is, which of these people has knee pain? The person on the left with a wide open joint space, i.e. Intact cartilage on both sides of the bone, or the person on the right that one side of their joint space has been entirely obliterated? That's what the answer is supposed to be, but if the answer was the person on the right, I could sit down and I wouldn't have to tell you anything about pain and pain processing. As it turns out, this is by far the most common cause of arthritis in humans, so-called osteoarthritis. It's the type of arthritis that most of us will get if we get old enough. The rate of osteoarthritis is going up exponentially in the United States as we get older and fatter as a country. The estimates are now that about 50% of individuals will develop osteoarthritis just of their knee. That's not any other joints or any other areas of the body. Just given our lifespan, obesity, and some of these other factors. Now, as it turns out, the person on the left is the one that has severe pain, and the person on the right is the one that has no pain. And in fact, this is aligned with population-based studies. If you don't happen to be a pain researcher or a rheumatologist, you would not know that 30 to 40% of the people in the general population that have x-rays looking like the x-ray on the right, bone on bone, grade 4 radiographic evidence of osteoarthritis, have absolutely no pain. So, and 10 or 20% of people that have severe knee pain have entirely normal radiographs. So, what has become increasingly clear in the pain field is that this inordinate focus on the periphery and this attempt to explain most, if not all, pain on the basis of peripheral nociceptive input. And what I mean by peripheral nociceptive input means that there's a sensory nerve out in the periphery, or nerves that are being irritated either by chemicals, by mechanical pressure, or some other signals to send an impulse to the brain to be felt as pain. The reality is there's actually not any chronic pain condition where anything we can measure out in the periphery, an x-ray, an MRI, a cystoscopy, a laparoscopy, accurately predicts who's going to be experiencing pain or how severe the pain is going to be. The problem, and I'm not here to talk about this, is that our diagnostic paradigms, even at present, still assume that if someone has pain in the knee, that the problem is in the knee. If they have pain in the hip, the problem is in the hip. And they don't in any way account for, by far, our most common pain conditions, headache, irritable bowel syndrome, fibromyalgia. We know there's no problem in the periphery. The headaches aren't because there's brain tumors or problems out in the head. they likewise have central nervous system mechanisms, as I'm going to talk about in the next half hour or so, are contributing to most chronic pain states. And some of this, in fact, can be triggered by different types of stress. Now, in the pain field, one of the unfortunate things that happens in general in medicine, has certainly happened in the pain field, is when we see these individuals where there's a disparity between what we can see on an X-ray or an MRI and what they're experiencing, we've had a historical tendency to say there's something wrong with the person. It's psychological factors, they're crazy, it's all in your head. And a lot of times when I give this talk to pain audiences now, I say it really is all in their head because the advent of brain imaging has made it clear that there are neurobiological reasons by which people develop these chronic pain conditions without any peripheral nociceptive input in the periphery. These are not just operative in conditions like migraine and tension headache, irritable bowel syndrome, fibromyalgia, these very prevalent conditions, but now what we're realizing is these are playing a role in any chronic pain state. Low back pain, osteoarthritis, rheumatoid arthritis. At the end of the day, there are two systems that are sensing pain in the body. One of the systems in the body is sensing peripheral nociceptive input. Is there damage or inflammation out in the periphery? But that information, once it enters the central nervous system, gets filtered by the spinal cord in the brain, and the spinal cord in the brain can either turn down the volume control in that, so those individuals that have something really wrong with their knee but don't have any pain, have very robust descending analgesic activity in their brain such that even though there's something wrong in their knee, that information is not allowed to go up to the brain to be felt as pain. But at the other end of the bell-shaped curve are individuals that have a very high volume control setting in their brain and their spinal cord, and they don't have to have anything wrong in order to have pain diffusely throughout their body, as occurs again in conditions like fibromyalgia. So what I'm going to do is talk a little bit about what we learned about the biology of these conditions, the treatment of these conditions, and then highlight the role that stress may have in leading to these conditions. Now I want to emphasize that these are non-psychological, non-psychiatric factors that seem to turn up and turn down the volume control setting, that these are somewhat or largely independent of constructs like depression, PTSD, and other psychiatric disorders that have been talked about more today. So the direction that we need to go is instead of saying that someone has osteoarthritis or rheumatoid arthritis or cancer pain or neuropathic pain or fibromyalgia, what we instead need to move towards is where we actually identify the underlying mechanism in each individual, because what we know is that although most individuals with osteoarthritis or cancer pain or rheumatoid arthritis do have ongoing peripheral nociceptive input, i.e. there is something wrong out in their joints, out in their tissues, we know that about 30% of people with osteoarthritis or rheumatoid arthritis or cancer pain have so-called centralized their pain, and that the central nervous system is playing a very active role in their pain, akin to what you would see in someone with fibromyalgia that doesn't necessarily have anything at all wrong out in the peripheral tissues. So I'll talk about fibromyalgia for just a second, more as a metaphor rather than a standalone diagnosis, because fibromyalgia is the first pain condition that in general we realized must be coming from the brain rather than coming from peripheral tissues. But every year or two, there's a new pain condition where we realize the brain is playing a more prominent role than the peripheral tissues. And as that occurs, we actually change the name of these pain conditions. So fibromyalgia used to be called fibrositis until we learned there was no itis. Itis means inflammation of the tissues, and we changed the name to algia, which means that there's pain. Irritable bowel used to be called spastic colitis. I now lead a NIH network studying interstitial cystitis, a chronic pelvic pain condition, and that within the next 6 months to a year will be renamed bladder pain syndrome instead of interstitial cystitis. So what's happened over time is that the initial idea always is peripherally focused, that there's inflammation out in the periphery that's causing these different pain conditions, but the reality is most pain, until people start to get older and develop wear and tear conditions like osteoarthritis, or if they have an autoimmune disease, which only occurs in about 1% of the population, like rheumatoid arthritis, they can have pain on the basis of peripheral nociceptive input. But if you look at people before they get to be age 50 or so, by far the most prevalent pain conditions are these central pain conditions: fibromyalgia, irritable bowel syndrome, headache, endometriosis, where we're learning with each advancing year, we're learning that these are more a brain and a spinal cord pain amplification problem rather than a problem out in the periphery where the pain is actually experienced. And so our group has spent a lot of time moving from thinking of fibromyalgia as a discrete or unique illness to more of a final common pathway, that all it really is that these people, that the central nervous system is playing a very prominent role, and in fact the only role in fibromyalgia, but fibromyalgia patients have more severe and more widespread pain than any other rheumatologic condition that we actually know of, even though the pain doesn't seem to be coming from the periphery. Now, there's a lot of overlap between these regional pain syndromes or widespread pain syndrome and psychiatric disorder, and we think that one of the big reasons for this is they share some of the same brain regions and share some of the same neurotransmitters, and I'll talk more about that. But in clinical practice, we're learning that the simplest way to identify whether people have a more peripheral reason for their pain or a more central reason for their pain is ask them pretty simple questions. Give them a body map and ask them to mark in different areas of the body where they have had chronic pain over the course of their lifetime. And the more X's that a person has, the more likely that they have a spinal cord or a brain problem, because what will almost always happen with people that end up developing fibromyalgia at, say, age 35 or 40, they're finally diagnosed as fibromyalgia. Those people start out in adolescence having functional abdominal pain, painful menstrual periods, headaches, and that's just the start of their pain, and then their pain becomes more widespread and more severe as they get older, and then they finally get labeled as having a condition like fibromyalgia. But you can almost always track going into childhood that these individuals are more pain sensitive throughout their entire lives, and I'll show you some data suggesting that genetics is playing a very strong role in pain sensitivity. Just in the last 5 years or so, 5 or 6 different sets of genes have been identified that are capable of turning up the volume control setting such that if you happen to be born with 1 or 2 or 3 of those genes, you will have experienced more pain for the same amount of nociceptive input. And if your volume control is really high, it's like a radio that is set to no station with just static. This is what seems to be happening in conditions like fibromyalgia, is that there's no filtering that occurs in normal individuals, and people get barraged with pain and other sensory information because so many of the filters that are present in normal individuals are not present in these individuals. Now, the way we can identify these people is they don't just have pain all over their body, they have fatigue, memory problems, and sleep disturbances also, either at the same time as they're experiencing pain or at other times during their lives. And again, by far the most plausible, biological plausible reason that this occurs is the same neurotransmitters that are capable of turning up or turning down the volume control setting in pain in the spinal cord, control sleep, control mood, control level of alertness. These are things like glutamate, GABA, substance P, norepinephrine, serotonin. So if someone has low serotonin in one area of their brain, that might turn up their volume control for pain processing. In another area of their brain, that may make them sensitive to all sensory experiences, not just pain. And in yet another area of the brain, that might cause them to sleep poorly or develop a mood disturbance. So what we're learning is that some of these critical neurotransmitters in neuroscience is like real estate. What's most important is location, 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 is that the neurotransmitter imbalance in one area of the brain might lead to one clinical phenotype, where that same neurotransmitter imbalance in another area of the brain leads to a different clinical phenotype. But there's only about 6 or 8 neurotransmitters that the body uses to send signals between all neurons, and at the end of the day, some of these neurotransmitters have systemic influences, genes that will make serotonin low everywhere in the body, GABA low everywhere in the body, glutamate high everywhere in the body, and this leads to a lot of these sort of chronic somatic symptoms and pain states. But again, it's not just pain. Chronic fatigue syndrome is another condition that's squarely in this entire spectrum of illness, where people have pain as a more minor complaint, but fatigue is their major complaint rather than pain. These illnesses occur about 1.5 to 2 times more common in women than in men. Again, there's very strong familial and genetic underpinnings, and the reason I'm here is that these can be triggered or exacerbated by a variety of different types of biological stress. Now, I can't help myself. Intermittently, I'll lapse into being a pain researcher instead of a stress researcher because that's sort of what I've become. But one of the exciting things in the pain field is that the exact same pain-prone phenotype predicts everything bad in the pain field. So we are always vexed why does such a small proportion of people with acute low back pain develop chronic low back pain? Why does a small percentage of people that has any surgical procedure develop new chronic pain at the site of their surgical procedure? But it turns out the exact same phenotypic traits predict everything bad in the pain field. The transition from acute to chronic pain, new chronic pain after a surgical procedure. And so there's been a lot of work looking at this so-called pain-prone phenotype, as well as trying to identify the genotypic correlates of this pain-prone phenotype. Because what seems to happen is the pain-prone phenotype is female, genetic factors, early life trauma, a family history of chronic pain and mood disorders. It turns out that chronic pain is one of the most familial disorders that we know of in medicine, but this was only realized within the last 5 to 6 years or so. And then cognitions like catastrophizing, and then some of the things that we can measure on an experimental basis, brain imaging, experimental pain testing. But the people with this phenotype, when they're exposed to different types of stress or acute peripheral nociceptive input, this can be modulated by psychological and behavioral responses to that pain or stressor. These are the people that are at highest risk of developing a new or different region of chronic pain. Now, the stressors that seem to be capable of triggering these illnesses, a couple of them I'll go into much more detail, are early life stressors. Having a peripheral pain syndrome, I alluded to this earlier, but about 20 to 30% of people with rheumatoid arthritis, osteoarthritis, or lupus, our classic rheumatic diseases, develop comorbid fibromyalgia or comorbid centralized pain. And when that occurs, they stop responding to peripherally directed orally administered analgesics or peripherally directed therapies like surgical procedures or injections, and we have to move more towards treating those people like they're a fibromyalgia patient rather than a osteoarthritis patient. Physical trauma can trigger this, like motor vehicle accidents. War can trigger this. Our group spent a tremendous amount of time studying post-deployment syndromes, the first Gulf War, these current wars. And in the first Gulf War, there was an epidemic of fibromyalgia and chronic fatigue-like illnesses after the first Gulf War. After these two current wars, all three of these things are occurring. Severe PTSD, mood disorders, and these pain syndromes are occurring in part because the amount of stress that people are exposed to in the current wars is significantly greater than it was in the first Gulf War. There was almost no PTSD after the first Gulf War because you seem to need to have very vivid, personally traumatic stressors in order to get PTSD. And the way we fought the first Gulf War, there was virtually none of that and no PTSD. So the nature of the conflict, most of you will know that PTSD was invented after the Vietnam War. The nature of war will determine the relative rates of PTSD versus depression versus these somatic syndromes. And after the first Gulf War, we saw almost entirely the somatic syndromes without depression or PTSD. Infections can trigger this, and psychological stress or distress can trigger this. Now, for those of you who may not be familiar with a condition like fibromyalgia, I will just tell you, as someone that spent much of my life and my career studying it, no one used to believe that fibromyalgia was a real illness. They used to think that these were just neurotic middle-aged women. There's still some people that think that, but those are people that will never take their head out of the sand and look at data and look at evidence, so I've stopped talking to those people in the last couple of years, because there's overwhelming evidence and overwhelming data that there's objective evidence of pain amplification in these conditions like fibromyalgia, irritable bowel, and across the entire spectrum of pain disorders. But it's sort of been dogma in the pain field for a long time that conditions like fibromyalgia were due to stress, were triggered by stress. But I just want to point out that actually is largely wrong. It seemed like the perfect story because some of the early studies in fibromyalgia done at the NIH before I started working with George, these were studies that were done by Leslie Crawford and Mark Demetrak in chronic fatigue syndrome. showed HPA abnormalities in people with fibromyalgia and chronic fatigue syndrome. But as those studies went on longer and longer, there were inconsistencies in the direction of the abnormalities, probably because there were inconsistencies in the rates of comorbid PTSD and comorbid major depression versus atypical depression in the individuals who were being studied. Also because in subsequent studies that we did, looking directly at CRH in the spinal fluid, the most potent driver of CRH in the spinal fluid is how much pain someone was in. So pain is a driver of stress. Anyone that takes care of postoperative people know that the most common cause for hypertension after a surgical procedure is pain. Treat the pain and the blood pressure will usually go down. And so a lot of those studies that were done looking at the HPA axis in conditions like fibromyalgia and in pain states were probably tremendously confounded by all the other things that occur, the early life trauma, the psychiatric comorbidities, and the other things that occur very commonly in the pain states. There's a lot of evidence that different types of stress, when people are exposed to those stress, for example, the terrorist attacks of 9/11, we accidentally did a study in Washington, D.C., where we were having fibromyalgia patients wear Palm Pilots that were beeping 5 times a day and tracking their symptoms. Right in the middle of this 12-week study, the planes hit the Pentagon, and the planes, we were still at Georgetown then, and the planes hit New York City. And there were accidental studies that were being done by our group in Washington, D.C., and by Karen Raphael in New York City that showed that after the stress of the 9/11 attacks, there was no increase at all, either in fibromyalgia patients or in the general population, in pain, fatigue, or in stress per se. Perhaps because the nature of the stress, if any of you happen to be living in Washington, D.C., or New York City at the time, it was even more horrifying than living in the rest of the country because there were tanks going down the streets. But the one thing that occurred immediately after those attacks is this incredible sense of patriotism and this coming together of the country. So people didn't experience that type of stress alone on an island like you do getting in rush hour traffic, get in an argument with your spouse or your boss. And the context of stress, whether it's characterized by a sense of control, a sense of support, may have a lot to do with whether it actually triggers illnesses like this or whether the stress is actually sort of good stress that actually makes people feel better. But in the pain field, for a long time, there were retrospective reports that early life trauma was associated with chronic pain in a number of different chronic pain cohorts, but it wasn't until a couple years ago when UK researchers exploited what's called the 1958 UK birth cohort. This is an incredibly cool study that the United Kingdom did in 1958, that they decided in 1958 that they wanted to follow every single person born in the UK in 1 week in 1958 for the rest of their life. And they now are still actively following 71% of those people some 50 years later, or I guess it's 53 years later because I was born in 1958. What they found is that they went back and they found that at age 8, the children's parents were sent a survey asking about different bad things that might have happened to the child. Excuse me. If the child had pain at age 8, the likelihood that they had fibromyalgia 42 years later was 1.5 times greater. If the child was in a traffic accident at age 8, the likelihood that they had fibromyalgia 42 years later was 1.5 times greater. You can see all of those life stressors increase the odds ratio of developing fibromyalgia 42 years later by 50 to 100%. And if you had a bunch of bad stuff happen to you in that year, your risk could have been 2.5 or 3-fold greater. So this is the only prospective data showing that early life trauma at age 8 is a very potent primer or trigger of developing chronic pain later in life. And again, this didn't at all need to look at retrospective recall. Now, that's psychological stress. The problem, though, is every kind of biological stress seems to be capable of triggering this spectrum of illness. A bunch of different infections, Lyme disease, urinary tract infections, infections of the vagina are all known in about 5 to 10% of people to trigger a new regional pain condition in individuals. And The things that trigger widespread pain are things like an Epstein-Barr virus infection or Lyme disease. The things that will trigger interstitial cystitis are repeated urinary tract infections. Vulvodynia or chronic vulvar pain will be triggered by sexually transmitted diseases. So it seems like about 5 or 10% of people that have an infection that causes usually subacute pain in an area of the body, that about 5 or 10% of people will never become pain-free, that they will develop chronic pain as a result of that infection rather than just have an acute self-limited course. And by far the best study looking at this was a study funded by the CDC in the Dubbo region of Australia. What they did is they followed people who were seen by primary care physicians with 3 totally different types of viral infections, Epstein-Barr virus, Q fever, and Ross River virus. Those cause totally different acute and subacute manifestations. But what they found is with those 3 viral infections, I guess one of them is not really a virus per se, that the same exact factors, this pain-prone phenotype, predicted which of those people, the 5 to 10% of people, would go on to develop a chronic fatigue-like fibromyalgia picture. And the exact same proportion of all 3 of those infections developed this chronic fatigue-like picture. And it had nothing to do with cytokine profile of the acute infection, with clearance to the pathogen, with baseline psychological factors. It was all this pain-prone phenotype that I showed you earlier that predicted the 5 to 10% of individuals who were going to go on to develop a chronic pain state like fibromyalgia or chronic fatigue syndrome rather than having an acute self-limited course. So what we seem to be dealing with here is that people are born at different points in this bell-shaped curve of pain sensitivity. I labeled this tenderness here, but it's a synonym for pain sensitivity. Where you're at on this bell-shaped curve is about 50% the genes you're born with and a 50% environmental things that happen to you. But early life trauma and stress can clearly move people to the right side of that bell-shaped curve. As it turns out, not sleeping well and not exercising regularly also move you to the right side of that bell-shaped curve. So 2 of the most effective treatments for these central pain states besides drugs are getting people sleeping better and getting them exercising well. That probably sounds familiar to some of the psychiatrists that treat mood disorders or PTSD. Again, we knew that these conditions like fibromyalgia and temporal mandibular disorder and irritable bowel and headache were all characterized by augmented pain processing in the central nervous system, but it's only been the last 3 or 4 years that we've appreciated that all chronic pain states, that there's a proportion of people that so-called centralize their pain that look identical to fibromyalgia, and that once that occurs, these individuals have to be treated with different drugs and different non-drug therapies than people with a purely peripheral or neuropathic pain state. I alluded to the fact that genes are being shown to play a very important role. If you care, here are some of the genes that have been identified. And literally all of the genes that have been identified seem to be either turning up or turning down the levels of these different neurotransmitters. And again, the psychiatrists that study mood will look at these neurotransmitters and say, I know these neurotransmitters, these are the same ones that I'm dealing with. But the reality is that the neurotransmitters that increase pain sensitivity are substance P, glutamate, and other excitatory amino acids, and nerve growth factor. Serotonin is a little bit schizophrenic depending on what receptor it binds to. It can either increase pain sensitivity or decrease pain sensitivity. In general, we think of norepinephrine, serotonin, and dopamine as being anti-nociceptive or inhibiting pain, as we do GABA. And even though we think of the opioid system as being an endogenous analgesic system, these arrows here are the direction of the abnormality in these neurotransmitter systems in people with fibromyalgia. And the only neurotransmitter system that's been studied to date in fibromyalgia that has not been abnormal in the direction we would have anticipated. We would have anticipated that a bunch of these would be elevated and a bunch of these would be low, and that would shift people towards being more pain sensitive. But the endogenous opioid system in fibromyalgia patients has actually increased activity, and PET imaging using a mu-opioid tracer has shown that fibromyalgia patients actually don't have hardly any unoccupied mu-opioid receptors in their brain. So this helps explain why opioid analgesics, i.e. narcotics, don't work in these centralized pain states. In fact, a lot of us think that not only do the opioids not work in conditions like fibromyalgia, but that people with fibromyalgia are at high risk for developing so-called opioid-induced hyperalgesia. We're giving those people an opioid will actually turn their volume control up higher and higher, make them more pain sensitive, and actually make them worse in front of your eyes. And in those individuals, the way to treat their pain is first get them off of the opioid, and then go on and try to actually manage the hyperalgesia that might have occurred for some other region. There's a lot of evidence that this isn't just a problem with pain processing. People with fibromyalgia and other pain states are sensitive to the brightness of lights, the loudness of noises, odors. And Rick Harris in our group has done really elegant studies honing in on the anterior and posterior insula. The anterior insula is involved in the affective regulation of pain, whereas the posterior insula is more the sensory intensity of pain. And what he's shown is that the glutamate levels are high in the anterior and posterior insula. The GABA levels are low. So what we're doing is sort of increasing the excitability of these brain regions. And he's shown that with drugs like pregabalin, which is a drug Lyrica, there's very clear evidence now from Rick's imaging studies that Lyrica is working by reducing glutamatergic activity in the insula that reduces functional connectivity between the insula and a number of networks, including the default mode network, and you can see which people are going to respond to the Lyrica at baseline because it's the people who have the highest levels of glutamate in the insula that are the ones that are most likely to respond to a drug that seems to be acting on glutamatergic activity. So functional imaging has taught us a tremendous amount. Much of what I'm talking about today has been in the last 10 years since the advent of functional neuroimaging and what we've learned about pain processing. We've learned that this increased volume control is really seen in a broad variety of chronic pain states, that this is quite different than depression. Some of the brain regions, anterior cingulate, hippocampus, amygdala, are co-activated in people with pain plus depression, but those brain regions are usually not any more active in people with pain without major depressive disorder than in people without pain or without major depressive disorder. So it's when they have that comorbidity that we see these brain regions that are involved in affect start to become more activated. And then cognition's how people can think about their pain via the same kinds of prefrontal regions that people have talked about today in being involved in mood. These are all involved in affect as well. I mentioned this notion of connectivity. I know that this is a big area in the depression field. It's a big area in the pain field. Our group and another group at Harvard last year showed that that measures of connectivity between brain regions are very abnormal in chronic pain states like fibromyalgia. And what we've shown in a couple of subsequent studies, again done by Rick Harris and Vitaly Napadow at Harvard, is showing that the strength of the connectivity between the insula and brain regions that it's not normally connected to is highly related to someone's spontaneous ongoing pain. So in the pain field, the holy grail has been a biomarker for pain, something that we could put someone in a scanner and say, you know, we want to really know how much pain this person is experiencing in case they're lying to us or malingering or whatever. And 3 or 4 studies now suggest that the connectivity between brain regions might be such a biomarker, either measured by EEG or measured by functional MRI. And again, people like Rick have done a lot of work showing the default mode network, the executive attention network, are very impacted when people have chronic pain, and that this seems to be driven in part by glutamatergic activity in different brain regions. Same kinds of brain abnormalities with respect to the size of the brain that have been identified in depression and mood disorders and PTSD have also been involved in pain. I don't know what these mean. I don't think this really means that this is neuroplasticity because this changes so rapidly in the pain field. There have been studies showing that this can change within weeks. And so I don't think this is really brain atrophy per se, but I do think that we are seeing neuroplasticity. I just want to go back now to our classic osteoarthritis of the knee, this condition that until 3 or 4 years ago we thought was the classic peripheral pain syndrome, that there was something wrong in someone's knee. That information was sent via the sensory nerves up to the brain, and that's what osteoarthritis of the knee was. There was a lot of evidence that should clue us into the fact that that wasn't the case, the fact that there's such a big disparity between structure and pain. Also because some of the drugs that we think of as being peripherally acting drug, non-steroidal anti-inflammatory drugs, acetaminophen, opioids are probably working both peripherally and centrally, but these drugs don't work nearly as well as we used to think they did if you look at some of the latest Cochrane meta-analyses. But now there's been an explosion of knowledge in the last year or two showing that all of the same imaging findings and experimental pain testing findings that have been identified in conditions like fibromyalgia, irritable bowel, tension, headaches are being identified in osteoarthritis, rheumatoid arthritis, low back pain, at least subsets, these 30% or so of people that seem to be at risk of centralizing their pain because they have this pain-prone phenotype. And one of the biggest things in the pain field was the revelation that duloxetine or Cymbalta worked well for chronic low back pain and osteoarthritis of the knee. So it was approved by the FDA for chronic musculoskeletal pain. It doesn't matter one bit whether people are depressed or not. They are just as likely to respond to duloxetine or Cymbalta, which was originally developed as an antidepressant, if they are non-depressed as if they are depressed. So it really does not matter whether they have comorbid depression, and that's the case with other drugs like tricyclic drugs that were originally developed as antidepressants. I do this partly as a joke, but our group does a lot of collaboration with a lot of people in different fields. And whatever you want to call the spectrum of illness, I don't use the word fibromyalgia much anymore because some people will always view that as the F word and hate the term fibromyalgia. But whatever you want to call the spectrum of illness, it's everywhere, it's ubiquitous. And if there was a name for it, I think it would be by far the most common medical problem because these people have severe pain, fatigue, memory problems, sleep disturbances, and each medical subspecialty has one or two names for these people. They call them idiopathic. They say there's nothing wrong with them, but when you start looking into the brain and you start looking, it's the same thing over and over again, this pain-prone phenotype, the same set of people that you really see in all of these different conditions. So the direction we're going in the pain field is to acknowledge that knowing the disease someone has, like osteoarthritis or rheumatoid arthritis, doesn't really help us treat their pain because all pain states at some level are mixed pain states with, in an individual, peripheral factors might be playing a role, nerve factors might be playing a role, central nervous system might be playing a role, and depending on which of these are playing a role, markedly different drugs work. Opioids and nonsteroidal anti-inflammatory drugs don't work at all for conditions like fibromyalgia, irritable bowel, some of these other conditions. They work pretty well when there is peripheral nociceptive input, like in osteoarthritis or rheumatoid arthritis. Anti-inflammatory drugs work great in rheumatoid arthritis or lupus, but they don't work at all in conditions like fibromyalgia that are not characterized by inflammation. So this is where we are in the pain field now, is that the choice of drugs is really driven more by what category of pain people have rather than by the disease that people have. And then finally, I just want to say that non-drug therapies can be extremely effective. Normally, when I have longer to talk about pain, I go on a rant about this because things like education, exercise, cognitive behavioral therapy can be extremely effective in treating chronic pain. Our group, led by Dave Williams, developed a website that we tested, and we showed that this website alone that gave people education, exercise, and cognitive behavioral therapy was just as effective as any of the approved drugs for fibromyalgia. I'm not suggesting that people use this instead of the drugs. I'm suggesting, just as is recommended in treating any chronic medical illness, is that you use drug and non-drug therapies together because these non-drug therapies can be pretty effective. So to summarize, a variety of different types of stress, but not just psychological stress, seem to be capable of triggering chronic pain and fatigue states. These include early life trauma, physical trauma, ongoing peripheral nociceptive input like rheumatoid arthritis, lupus, infections, war, psychological stress. These central pain states are extremely common in the general population because they are called different things by different subspecialties. The current thinking is that some of these neurotransmitters, these shared neurotransmitters, are driving the fact that people have comorbid pain, fatigue, sleep disturbance, and mood. The trial that was done in the setting of fibromyalgia that was most illustrative of this is the drug gamma-hydroxybutyrate, which unfortunately is GHB, which is the date rape drug, but it is a very potent GABA agonist. It raises GABA levels. That drug, when administered to fibromyalgia patients, led to a simultaneous improvement in pain, in sleep, and in fatigue, and those were not related to each other. It seemed to be independently affecting those three domains because GABA it controls your level of alertness, your level of sleep, and your level of pain. And we see this with some of the other drugs, the serotonin, norepinephrine reuptake inhibitors. When we use those to treat pain, we make mood better, pain better, fatigue better, rather than making pain better. So one of the advantages, if you will, of treating these central pain syndromes rather than peripheral pain syndromes is people usually have more than pain as a symptom, and when our drugs and our non-drug therapies work, we'll usually see amelioration of symptoms other than just pain, some of these other comorbid symptoms that travel very closely. Thank you.
Thank you very much, Dan. We have time for questions, and actually questions can also be directed towards earlier speakers. Yes?
I'm speaking strictly as a layperson. Why then is the medical field really pushing morphine for all types of pain if it's known that it doesn't affect most types of pain?
How long do we have here? There's a couple issues. Number one is that narcotics were approved for pain before the FDA was in existence. So the rules that apply to a drug company that has a narcotic on the market are different than the rules that apply to a company that has a new chemical entity that began testing after 1962. So most opioids that get on the market get a broad label for chronic pain because that was what the labeling for opioids were before the FDA came into existence. That's the first problem. So the pharmaceutical companies are incredibly good at convincing people of things. That's why most of you know about fibromyalgia, because most of you watch the Lifetime Network or whatever. When I watch sports on TV, like I watch the news and sports, and my wife watches like chick TV. I have never seen a Cymbalta commercial on the TV that I watch, but if you, when I sit down and watch television with her, all the fibromyalgia commercials, all the Cymbalta commercials are aimed appropriately at TV that more females watch, because the women with chronic pain are the ones that are more likely to have a central nervous system component of their pain, and in fact more likely to respond to a drug like Cymbalta. So it's the pharmaceutical companies are pushing opioids. Most physicians have become anti-opioid, but one of the other problems is, starting about 10 years or so ago, there was a very active movement by JACO, the accreditation agency for hospitals and clinics, to start tracking pain and to following pain more closely. And people could even be fined if they didn't track and attend to people's pain on the inpatient setting. So what happened is we taught people they had to ask about pain more and treat it more aggressively, but our drugs didn't get any better. So that we still had the same drugs out there, and people were always taught that if the pain was bad enough and refractory enough, opioids would always work. And that just simply isn't the case. Again, there's a tremendous number of pain states for which opioids don't work at all, and in fact may actually be making people worse. The FDA knows that. The FDA is extremely concerned about that and trying to get opioids controlled a lot more so than they are right now.
David.
I agree with literally everything you said, and it's very important for the treatment of people with chronic and acute pain. I was surprised by one thing, though. In the non-pharmacologic treatments, you didn't mention hypnosis, which is the oldest and most empirically validated central treatment for pain, and I wonder if you could comment on that.
Yeah, again, it was because that wasn't really what I was supposed to talk as therapy. So I'm a huge advocate of a whole host of non-pharmacologic therapies. We've done a lot of work on acupuncture, acupressure. We have ongoing trials of CBT, adding emotional disclosure and things. So, yeah, I agree that there's yoga, Tai Chi, just so many non-pharmacologic therapies. But the problem is, I hate to say this, but no one's making money off of most of those things. I'm sorry. Okay. The market forces in the medical field are the drug companies, the large hospitals like the ones that I work for, that maybe you work for, that they make a lot of money on procedures, so there's no market force driving the adoption of these non-pharmacologic therapies that are not covered by third party. Often people are paying out of pocket for them, and that's why way too many people are getting injections and blocks that cost $1,000 or $2,000 and have never been shown to work very well, and not enough people are getting someone sitting down and talking to them, listening to them, or using some of these non-drug therapies. Okay, thanks.
Thank you very much. What do you think, Carol? Should we stop and... Okay, we're available for general questions. Dr. Gold, Dr. Clow, if anybody has any questions, please. Well, it was a very exciting and intensive day today. A lot of new knowledge. Actually, I was very glad to hear new things about fibromyalgia, especially that it's not called fibromyalgia anymore.
It's still called fibromyalgia.
How common is it, just in the community?
Using the old criteria, 2 to 4% of the U.S. population. Using new criteria that don't require doing a tender point exam, more like 6% of the population. Yeah, I doubt that my voice needs a microphone.
So 6% is a very high percentage.
But yeah.
There are many millions of people, you know.
Oh, yeah. Probably 15, 20 million Americans. And the notion that this is a disease of industrialization is been really blown apart. The rates are at least as high in developing countries. Another thing you don't know about me since we last saw each other, I spent a month in Kenya every year. And in Kenya, there's very high rates of chronic pain. So this notion that chronic pain is a lot due to worrying and, you know, again, the notion that fibromyalgia used to be yuppie flu. It was middle-aged women, you know, affluent. All that's really been blown apart by the epidemiologic studies in this spectrum of illness.
So in Kenya, it's just as frequent as it is here.
At least as frequent as it is here.
Yeah.
I don't know why. But.
Dr. Romero.
Could I ask you, what percentage of you referred to genetic markers to identify the patient that is prone to pain. What proportion explains the genetic component or the variance in?
About 50%.
And what are the candidate genes for this?
The strongest one that's been identified so far is COMT, catechol transferase, which is breaking down catecholamines. As you probably know, that has been looked at in psychiatric disorders, and it might be responsible for some of the sex differences in some of the different psychiatric disorders, but it's not associated with any single psychiatric disorders per se. But it's one of the attractive genes because it's an estrogen inducible gene, so it might help us explain why women are more pain sensitive and more sensory sensitive.
Yeah, it's also a gene that is very highly expressed in the placenta, and is a mechanism by which catecholamines are deactivated and don't cross from the mother to the fetus. So it's a very interesting biological system.
Right. So there's there's a KCNS gene, a potassium channel gene. There's some sodium channel genes. There are GCH1. What does that stand for? I don't know. I'm blocking on it. But there's about 6 genes. But for these candidate genes that have been identified so far, the odds ratios for developing disease or causing people to be more pain sensitive are in the range of 2 to 4. Very strong individual genes. So, you know, when you look at, for example, genome-wide association studies for something like type 2 diabetes, where no single gene had an odds ratio of more than like 1.1 for causing disease. And the pain field, the pain field hasn't historically been very well funded, so there hasn't yet been an appropriately powered GWAS study in pain. But the candidate gene studies, the ones that people guessed would be abnormal, some of the ones that I've been talking about, have all had, 100 patients versus 100 controls, and replicated in several studies. So they're fairly powerful compared to the genes that are playing roles in other states. And some of them are genes that you would recognize in, like the serotonin transporter gene has been shown to be a risk factor, just as it is for mood disorders.
Mood disorders.
Right. So.
Dr. Smopoulos?
I had the identical question because I was particularly interested in the genetic variants, how many there are. I'm sure there are common genetic variants with very little effect. If you have any idea about the frequency of genes, and if you have any idea in terms of tissue cultures, what silences these genes. And then move on to cannabinoids and what's the effect of cannabinoids.
We don't know much about the first couple questions that you've asked. Really, the pain field is so, all this is so new in the pain field. This is really last 5 to 10 years. So in contrast to the depression field, where you've known that the brain is the problem for a long time, this is all relatively new. The cannabinoids, I have to be careful here. So I didn't have this on my disclosure slide, but I went to the University of Michigan in the 70s. So let me make that clear before I say what I'm going to say about cannabinoids. But in most countries other than the US, there are one or more synthetic cannabinoids approved for pain. So as a pain researcher, there is actually a lot of evidence and a lot of data that cannabinoids work for chronic pain states. I think the biggest reason that there is not a synthetic cannabinoid approved for use in the United States for pain is political, not scientific. Intermittently, there have been entire 4 or 8 year periods where you can't do any research on good effects of cannabinoids.
Well, they make people
So that
fat. They make people fat.
Yeah, well, yeah, but when cannabinoids are given for pain, and they're given orally rather than when people smoke cannabinoids, they get the munchies and they get high. But when they take them orally and there's a stable level of cannabinoids, they don't have anywhere near the side effect profile, if you will, that we think of with recreational use of cannabinoids. And so in states that have had medicinal marijuana laws, there are a fair number of people that are getting some relief of their pain with cannabinoids. Those of us who are physicians and treaters would much rather have a synthetic cannabinoid that we could prescribe, that we would know the dose to give and things like that. But the notion that the cannabinoids don't work in chronic pain is not really supported by scientific data.
Any other questions? Yes, sir.
Thanks. Could you talk about how the political aspects, particularly the classification of certain narcotics and other drugs and the restrictions that have worked to restrict research in this area and have impeded maybe making some progress?
I guess I started, didn't I?
Yep.
I mean, I think the thing that you need to understand that as a scientist, there's a number of different paths that we can choose to go down in our scientific career. And why would we choose to go down a path that intermittently could be totally blocked by someone deciding politically that all of a sudden this type of research can't be funded, or that there was another set of, it's hard enough to get NIH funding, but to worry then about the political climate from year to year in NIDA or in an institute that might be funding research along these lines. So I think that people just stay out of it because it's like, why do I need even more aggravation than there is innately in being a biomedical researcher, where the funding lines are now at 8 to 10%, at the NIH, and so that 9 out of 10 grants we write are not going to get accepted. Why would you ask for an even higher bar to jump over by devoting your life to studying cannabinoids and pain? So I mean, I hate to say it, but we all have options as researchers, and I think that you'd have to somehow be personally incredibly pro-cannabinoid, maybe in a bad way, in a recreational way, before I could imagine deciding that you're going to, say that that's what my career is going to be, and I'm going to be at the whims of, politicians that every so many years might decide that this is not something worthy of NIH funding.
Right. Yes.
This is for Dr. Romero. You talked about the second and the third trimester and fetal pain. Can you talk about it in terms of the first trimester?
So that is a difficult area because the concept of pain, and I am in front of an expert, so please take over at any moment, it requires not only that there is a stimulus, but there is the awareness of pain. And that is a very difficult thing to prove in a patient that you cannot talk and cannot tell you whether he feels or she feels pains or not. So we don't have any evidence. I'm hesitant to say that there is pain in the first trimester. I believe that the data that I show provide indirect evidence that occurs in the second and third trimester. We know from ultrasound observations that when we place a needle and the baby in the amniotic cavity moves the arm over the leg into the needle, immediately there is a response, but that may be a reflex. Whether the baby has pain or not in the first trimester, we do not know. No, the second and third trimester. And if you feel that.
Well, how soon is the pain system developing in the embryo? Is that known?
No. No, I totally agree.
All right.
In fact, until a child is able to talk, studying anyone before that is like looking at an animal model where we say we're measuring a pain behavior, which is what you are measuring a pain behavior, and you're inferring that it's pain just like a tail flick test in a rat, but that's all you can do until the child can speak to you. The best you can do is measure a pain behavior as a surrogate.
Yes.
So I had a question. Am I right to summarize your talk as saying there's maybe two kinds of pain, one that's peripheral and one that's central, and having all these different diagnoses of fibromyalgia and other kinds of pain maybe counterproductive?
That's pretty close. I'd say that there's three types. There's peripheral nerve pain, which is relatively uncommon, but if you have either damage to a nerve or compression of a nerve, that can cause pain. So that was that middle category that I didn't talk very much about.
Toothache?
No, that would be like sciatica. If you have a herniated disc in your back, carpal tunnel syndrome, when the median nerve in your wrist is pinched and you get numbness and tingling. So those would be examples of nerve pain where the nerve is involved. But by far the more common causes are either our peripheral or central. And yes, more and more we're realizing that the name that you apply isn't that important. And in fact, it's often confusing because people with fibromyalgia, they're seeing a rheumatologist for their fibromyalgia, they're seeing a gastroenterologist for their irritable bowel, they're seeing a urologist for their interstitial cystitis, and they're getting conflicting. They have pain in all different areas of the body and different diagnoses for the pain in different areas of the body, and they don't get one person telling them this is one problem for which this is the best treatment. They'll often be going to all these different subspecialists that are giving conflicting answers, actually.
So is that sort of yours and the expert's opinion, or is that pervading the medical field? I mean, it makes a lot of sense to me. What I'm asking you is how widely accepted and known is that sort of view?
It's like any other scientific knowledge. It takes a fair amount of time for it to diffuse into the medical community, and it's more difficult to make this diffuse into the medical community because we're talking about 20 or 25 or 30 different subspecialties that have to get this information to them, because that's how we usually learn, unless you're a generalist and you're a general, but most of us are learning from subspecialists of the same subspecialty. So it turns out that we do work with gynecologists studying chronic pelvic pain and urologists studying, and they're the ones that then have to go give the talks at those meetings to convince the gynecologists, the urologists, that this is more of a brain problem rather than a peripheral problem. But it's going to take, 10, 15 years before this knowledge, because this is fairly new information, fairly new knowledge. There's no one in the pain field that would disagree with what I'm saying, but there's a fair number of people in the general medical community that might still be quite unaware of this sort of paradigm shift.
Dr. Rosenthal.
You said that 30% of the people with the bones right against each other wouldn't have pain, and 20% of the people with the bones not touching each other would have pain. It still is a big difference between those two, in that 70% of the bones touching each other will have pain, and 80% of the ones won't have pain in the other one. So it's still of value. It just doesn't explain all the variance, I guess, is what I'm understanding.
Totally agree. Yeah,
Okay.
so nociceptive input is playing a role,
Right.
but in some individuals that nociceptive input seems to be incredibly dampened by the central nervous system, and they don't have pain when it looks like they should. And in other individuals, it's the opposite, where it's being augmented. So it's just that it doesn't, focusing on that as the only cause of the pain, which is what is often done, is a misguided venture.
Thanks.
Any other question? Dr. Brown?
Just quickly, in that context, do you know, can you say anything about why acupuncture works?
So we do a lot of work on acupuncture. When I say we, Rick Harris in our group, who's a junior faculty member, does the work. He has shown, the reality is in chronic pain conditions, acupuncture has been sort of iffy with respect to whether it shows efficacy or not, shows that it works. Some studies show that it does, some studies show that it doesn't. It works way better in acute pain. So it's much more, the studies are much more consistent. That you've heard about people getting surgical procedures in China with acupuncture as the analgesia. So, but again, I think that's because chronic pain conditions are more heterogeneous and a little bit more complicated. The other problem with acupuncture research has historically been what is a credible sham procedure. So some people say that when you put the needles, the acupuncturist will say, that if you put the needles, but you don't put them in the right location, you're still doing acupuncture. It's just not as good of acupuncture, that there's no such thing as a placebo or a sham needle insertion, that any needle insertion would engender some type of an acupuncture effect, but if you put them in the right location, it would engender more of an effect. So there's a big controversy, and it's about like being a Democrat or a Republican. If you like, I mean, the people are too polarized. They either say that it works, and they don't look at any of the data showing that it maybe doesn't work, and then other people are saying, well, it's just all a placebo effect. I would have to say that as a pain researcher, I think the placebo, you're going to hear about placebo tomorrow. In pain, the placebo is pretty powerful. There's a lot of medical conditions where it's not powerful, but just getting someone to believe they're going to get better, I think that's what healers have been doing for millennia, even before we had effective drugs, is we were talking, people that used to be called healers are now called physicians. We'd sit down with people, we'd talk to them, we'd convince them they were going to get better. And we were using the art of medicine, and we were using the placebo effect to our advantage. And I think that a lot of medicine now, there's no such thing as a placebo effect, because doctors don't talk to their patients, they don't spend any time with them, they don't. And so a lot of that is lost, but where people do get that is in the settings where people are doing CAM therapies, the acupuncturists. Those are the people that sit and talk to people for a long time and develop a therapeutic alliance with them, the way that physicians maybe 50 or so years ago did, the old family physician did. But now in the day of the 6 to 7 minute return office visit, there's not a lot of that time that's being spent with people. And I think that does make all of our therapies less effective. If we're not engendering the body's own analgesic properties, which are incredibly powerful, then we're disadvantaging all of our therapies if that's how we're doing that. So I don't know whether acupuncture works for chronic pain. I do know that if someone believes it works, about 40% of those people will get improvement with acupuncture. And it's very safe, and it doesn't cost very much.
I think we're going to hear it tomorrow, but I recently read Dr. Benedetti's book, and apparently with acupuncture you get, in imaging studies, changes that the same that you get with placebo. but there are some extra sites in the brain that are affected. So it's not just placebo, it's something else. And the same is true for cognitive behavioral therapy or other therapies. In other words, the placebo areas are involved, but some other areas are involved as well, additional. One more question, last question.
I was just curious if your research indicated that there was any kind of a correlation with obesity and pain.
That's a good question.
The current data are pretty clear that obesity contributes to pain of weight-bearing joints. So we've known that for a long time. There's emerging data that suggests that obesity in general might be associated with this kind of more centralized pain. The pro-inflammatory cytokines that are released by the adipose tissue may be driving a very mild, sort of generalized inflammatory state that can lead to nociceptive input. But that's still more hypothesis. What we know for sure is in the context of arthritis, for example, of weight-bearing joints, that a 10-pound weight loss will lead to, in most people, to a clinically meaningful improvement in their pain if their pain is in the knee or the hip. If it's in a joint that is not weight-bearing, that doesn't seem to make as much of a difference. But the question I think you're asking is more a general effect of obesity, and that question is still unanswered. I think, again, you were seeing a lot of intriguing associations with BMI over the course of the day today, and I'll bet that in the next several years we're starting to see some of that seeping into the pain field as well.
Does pain cause obesity?
Chronic pain patients are much more obese than non-chronic pain patients, but the reasons for that are, so I don't want to say it's just pain, but pain causes inactivity, inactivity causes obesity. A lot of the drugs we use cause craving and cause weight gain. It would be the same as asking the question, does depression cause obesity? I think the answer would be just as yes, but because of all these different reasons.
Well, yeah, I agree with you. I think people who are in pain, it may be harder for them to exercise. The drugs that we use, as you say, I've seen 20, 30 pounds put on, both with Lyrica and Cymbalta in selected cases. And the other thing is that sweet foods have like an opiate effect. So it may be one's own way of dosing oneself.
Right.
So.
Right. Thank you very much. It's a wonderful day.
This has been a presentation of the Library of Congress. Visit us at loc.gov.